Yeah, so very warm welcome to all of you to the workshop on modeling in biology and medicine 2020. We are super happy to be here and also that you are here to listen and discuss about exciting projects on the edge of mathematics, biology, computer science, physics, and also medicine. My name is Barbara, and together with Philip and Felix, we are the organizing committee of the workshop. And I think you can see them also with the videos on.
Hi.
We are PhD students at the Department of Mathematics at the Gothenburg University and the Department of Biology and Biological Engineering at Chalmers. So to start with this, I want to give a brief overview of this workshop. And it all started basically around 2 years ago when Philip, Felix, and back then also Johannes, we sat down and we wanted to organize a workshop for modelers related to biology and medicine, which didn't really exist at that point, at least not in Sweden. So we had the ambition to establish a national network. And in particular, since we are PhD students, we wanted to focus on young researchers that are in their PhD studies and postdocs, to connect them, to give them the opportunity to present, but also to encourage future collaborations. So we put all our forces together, and in 2019 we had the first workshop, which was a lot of fun, I think. And after that we were eager to repeat it, to kind of make it a thing. And then we started early, we tried to avoid all the issues we had the year before, and planned the second workshop for May 2020. But then unfortunately, because of the ongoing pandemic, we had to cancel it. But very quickly we decided that why not making a virtual workshop instead? How hard can it be, kind of? So we did that. We faced a lot of new challenges. So instead we had to test tools, we had to find out what we can do, what are the possibilities, what we want to do, and so on. Meanwhile, we also lost our fourth, Johannes, who recently became a father in the so he was not in the organizing team anymore, but he's here, I think, which we're also very happy about. We're happy in general that there's so many people joined today and followed our invitation, and we hope that it also stays a thing, so that there will be another workshop next year and hopefully also the years after. The program for these 2 days, it looks like that. So we have a bunch of talks now in the beginning, which all will be in this Zoom room that you're in right now. We will have a poster session in the afternoon now, where we have posters that are all in parallel Zoom sessions. So each poster will have an own Zoom room that you can join and leave freely as you like, and start discussions with the presenter. We have a social activity tonight, which will happen on Gather Town. And we would also be happy if you join there, because we have a little challenge for you in the beginning. Tomorrow, after the session or the closing, basically we have speakers rooms with all the invited speakers. So Jan, Lars, Gunnar, Falco, and Jeanette, who are basically available for asking more questions or having in-depth discussions, but not all the invited speakers will be there all the time. So we will give the information on Zulib, which brings me to the next point, because Zulib is our main platform for communication. You will find all the information there, you will find all the links there, and the program, and it's also a platform for you to chat and discuss. Zulib is, I don't know how many have experience with that, but it's organized in streams, and each stream has a topic. So here, for example, we have general information where you find all the general informations and so on. The posters where you find all the poster links to the Zoom session with passwords. We have the speakers rooms again with the Zoom links, and also the stream for the talks, and a tech support stream where you can ask questions in case there is anything that is not working. In the posters and the talks, you will find right now only the abstracts or maybe the Zoom link, but we would like to encourage you to use these topics there to ask questions, to discuss, before, meanwhile, and after the talk or the presentations. So use this to ask and discuss. Yes. Also, we would like to ask you that during the main talks, we use this stream talks as a main community, like a main platform for asking questions. So, hello. So if you have a question during a talk, please write it here, and then we will moderate through them in the end, in the question session of each talk. Yes, if you have any problem, if you can't enter a meeting, if you encounter anything that doesn't work, or you just want to give us feedback, you're of course welcome to contact us anytime at either Felix, Philip, or me. And this you can do via mail or Zulip, and I guess Zulip is the faster version. Also, we will try to be on this tech support. We will answer there as quickly as possible all the time. Yes, that's more or less it for now. We will have a recording now all the time, so if a speaker doesn't agree that we record this, please let us know in advance. Yes. If you are active on Twitter, we have a hashtag that you can use now during the workshop and promote the workshop. Yes. Are there maybe any questions on the organizational part so far? No? Good. Then I think we can actually directly move to the first speaker, which is our international guest. It's Jan Hasenauer from Bonn University in Germany. He received his PhD in systems biology, went some time to Munich at the Helmholtz Center afterwards before he came to Bonn, where he now leads a research group focusing on developing and applying computational methods for the analysis of complex biological processes, with particular focus on data-driven methods. And he describes his work by citing Galileo Galilei, measure what can be measured and make measurable what cannot be measured. We are very happy to have you, Jan, and please, just one small remark. I think 2 minutes before the talk or the time is over, we will try to give a little sign, which sounds like this. So we hope that it doesn't disturb the flow too much, but then you know what that means. Okay, thank you very much, and please, Jan.
Yeah, thank you very much for having me. Thanks a lot to the organizers for putting together this event in this very special and challenging times. And I really find it great that there are these possibilities now to network and to connect on these different levels. So thanks a lot to all of you. I know that this can be a lot of work. Let me start sharing my screen. I hope that you can see it. If not, please speak up. Okay, all right. I was struggling quite a while deciding what to talk about today, and I decided to go kind of for an overview on challenges we encounter in our research, approaches we have chosen, which might be transferable to a certain degree, and this all in the context of high throughput and high content data, because specifically nowadays, I mean, data is becoming more and more abundant, and it's sometimes difficult to find the best way to deal with this data, to start interpreting it, and to understand the underlying processes, basically. In particular, this whole topic of high throughput data is obviously nowadays completely surrounding us. Companies like IBM announced already more than 5 years ago that the era of cognitive health starting to use machine learning and other approaches basically to explore larger and larger data sets basically, and are still going with there are these big companies like 23andMe, which many of you I guess know basically, which perform relatively large scale studies nowadays, basically collaborate with different pharma companies, basically also having, for instance, Google backends for the different companies and so on and so forth, and performing these large scale genetic tests to inform individuals about potential risk factors and how they might want to adapt their personal lifestyle. But on the other side, also sharing this data with big pharma companies like Pfizer and so on, which want to use this for optimizing their development processes and so on and so forth. And these are really gigantic numbers and gigantic data sets we are speaking about. Already here, this was 3 years ago, basically, about data for more than 600,000 individuals was shared basically between these companies to explore new biology, to improve drug development and similar things, basically. And data sets are steadily growing. We are currently in a project where we where a partner is collecting Codex imaging data where a single image has 2 terabytes, basically. So quickly filling up even large data storage devices, basically. And very often it's still a bit unclear what to do with these data. This kind of brings me to the goal. So in my opinion, at least if we focus on the medicine side, usually the 3 aspects we are interested in, or one is interested in, is obviously diagnosis, prevention, and treatment. And for all of these different, let's call it application areas, there are different type of approaches chosen. If we speak about diagnosis, this is often related to so-called cohort studies, statistical correlation, association, risk markers, biomarkers, basically, and so on. In the context of prevention, it's more risk assessment and linking already to topics like optimal control and design of intervention and prevention strategies. And in the context of treatment, it's more about response prediction, basically, and drug mechanisms. And in all of these contexts, I think understanding plays a key role. And it was really shown that basic understanding of molecular processes can really enhance the different things. On the level of cohort studies and statistical correlations, power can be improved by not considering arbitrary links, but to focus on things which are biologically plausible. On the level of prevention, obviously, if you have a model to base your optimal control on, then this can obviously improve predictive power and so on and so forth. And so there's this whole topic of mathematical modeling, mechanistic, semi-quantitative, all of these different types of computational models, which can really contribute to these different areas. But there are many challenges, at least in my experience. There are quite a few walls one can hit, basically, if one tries to apply the different methods, basically, and try to answer some questions. Here are just a few things, basically, we encountered to be challenging. If we deal with large-scale models, with large-scale data sets, usually you also want to have large-scale models. And the formulation of these models is usually quite time-consuming. Basically, reusability of published models is an issue and so on. Then there's this whole topic of model simulation, which on its own, which is the basic way of evaluating a model, but already the simulation can be quite time consuming, in particular if it comes to models for spatial processes or stochastic simulations. Basically, individual simulations can take up minutes, hours, or in some cases, even days, basically. And then there's this whole topic of actually how to use a model. Very often there's a certain objective you want to reach, and then you want to formulate or evaluate an objective function, which can be tricky on its own because translating the idea one has into a formal description of an objective function is sometimes tricky. In particular, if one has many different data types which are present and which play a role, basically. And then there's this topic of parameter estimation, so training the model parameters with the corresponding data, assessing uncertainties, and even more tricky, model selection and model refinement. For small-scale models, these things are more or less established, I would say, although even there some challenges persist. But as soon as one looks at models with more than a dozen species and a dozen parameters, these things can become extremely tricky already. So what I would like to focus on today is a few of these things, some ideas, not necessarily from many of them, not from us, but from others, and some approaches I find interesting and things which might be valuable to think about, and obviously also some ingredients we have been working on over the last years. So the first question usually is how to get the model or models and potentially also the data sets if you don't have them. I will today mostly focus on the models, but obviously also collecting the data sets can sometimes be tricky. The simple answer to that is usually, yeah, we can develop it ourselves, this model. But if it comes to large-scale data sets and therefore also large-scale models, often resources are quite limited. It's very unlikely, or not too many persons succeed developing a high-quality genome scale model in a certain project. PhD project, postdoc project, or so on. Basically, this is sometimes simply not realistic. So the question is, what can one do instead if one faces with large scale projects? One thing is automation of the corresponding tasks, and there are nowadays quite a few really nice tools which have been proposed already a few years back. For instance, from a group of Peter Sorger, an approach to automatically construct executable models. Here in this case for signaling networks, but there are other approaches which are also now feasible for metabolic networks and similar things, just based on verbal description, basically, which can be found in the literature. So there's a lot of techniques, basically based on machine learning, automatic language processing, basically, which nowadays allow us to construct at least scaffolds of these models in a more or less automated way. One tool there is with Indra, which was proposed, but there are other groups intensively working on this topic. The issue with that is that, as I said, it's mostly still scaffolds, and getting a completely biologically plausible description of a process is extremely difficult, and at least from what I have seen, is not possible in a fully automated manner. But these things give you a very, very good starting point and can really accelerate the process. So what's usually still done if one faces with large-scale data sets is to start reusing models on one or the other side, and potentially also start reusing data sets. I mean, this whole topic of reusing things is obviously nowadays quite present. It's also related to this whole issue of reproducibility, which is, I think we can all agree on that, in many situations not ideal. But there are many, not only researchers, but nowadays also journals pushing for that, to improve that, basically. There are many studies of what can go wrong, basically, how it makes science less productive. And I mean, there's this whole question of also accessibility of research, hiding behind paywalls and things like that. So there's a huge drive towards this open and reproducible and long-term maintained models and data sets. So here, really data also in terms of models. Unfortunately, there are quite a few initiatives which you all know, like Combine, which contribute to that, developing standards and so on and so forth. But there are these resources, so delete the but, and by this related to all of these standards, there are these great resources like biomodels, JWS, and so on and so forth, which collect a large set of published models. But unfortunately, only a fraction of the models we have is uploaded, is available publicly. And there's, again, this huge problem of reproducibility. And I think this is really a gigantic issue. I mean, what we usually see is that individual studies focus on specific pathways and really ignore what's going on outside of the pathways. Sometimes this is a valid approach. However, in other situations, this can be super problematic. We all know about crosstalk, basically, which is very often disregarded, basically. And this is just hiding all of these influence factors outside of a pathway of interest, making predictions and so on, very problematic. And I think we can all agree that it would be nice to be able to fully appreciate and take this crosstalk into account and to consider that, basically. So we have to go away from looking at pathways in an isolated manner to reduce complexity and effort, but we, I think, could agree on that very often we have to really look at the full picture, basically, and have to integrate all these different things. What we are currently working on in a project is to try to build a community, and this is why I mentioned that here, basically. I mean, already having a community here is obviously quite nice, to bring together already available data sets, models, in a kind of a standardized perform and to be automatically able to integrate that basically into large scale models using automatic model merging tools, for instance, developed by the group of Olaf Wolkenhauer located in Rostock. And we started that already a few years back with a back then relatively small collection of models, back then only 20 models and the corresponding data set, which was still new in a sense that it provided, because the data sets were directly linked, more information than already available databases like biomodels or JWS, and it allowed us directly to look at these combinations, basically fitting problems and characteristics of the different models, basically like what properties do we have regarding initial conditions, noise models, basically. And this allowed us then to evaluate, benchmark different types of fitting tools, basically, and the performance of different algorithms. And what I'm going to present now in the following is a bit based on this collection, some experiences we made and benchmarking this, basically, and some, at least for us, surprising findings, basically, which some persons might not be aware of. So this brings me directly to a second question, basically how to inform models using from data, basically, so linking to this topic of parameter estimation, uncertainty analysis, and so on and so forth. And I'm now looking at a specific example, but many of the things directly generalized. So in my research, we are very often looking at ODE models, ordinary differential equations, where we have some description of dynamics. X is the, for instance, the concentration of a set of biochemical species, F, usually described as stoichiometric matrix times flux vectors, where some observables, what we observe are some noisy measurements of these observables. And we can write down a likelihood function, which you can interpret as a distance between the observed data and the model simulation, basically. And then usually a first step is to perform maximum likelihood estimation. You can also do Bayesian parameter estimation if you have a prior on the parameters. And you can use optimization sampling. I would like to focus in the first step here on the optimization. Just one common thing, multi-start local optimization, which is very often applied here. So here on the left-hand side, you see the corresponding data set, just an illustration with some error bars. Basically, this might be your model simulation for specific parameter values in the parameter plane. And how these methods work is that you essentially do iterative exploration of a parameter space, starting at some point, calculating, for instance, a gradient, improving the parameter, and then repeating this process over and over. And usually this is started from multiple starting points to actually assess reproducibility and so on of a fitting results. Multi-start local optimization is a common approach, but there are many other approaches which can be taken, and I will come back to that in a second, on a few minutes, I guess. The first question we were interested in when looking at this collection of models, basically, for this performance evaluation was, what do we actually have to worry about? I mean, if you are in a practical project, then there are many things you can worry about. So the question is what you should focus on. And we were evaluating a few claims which are made in literature to check whether we really find that for this set of models we looked at. And the first thing we tried to assess was the no free lunch theorem. So many persons in the audience might be aware of that. So this is a common theorem which is very often stated in the context of selecting optimizers. And it essentially said what an algorithm gains in performance on one class of problems, it necessarily offsets by its performance on the remaining class of problems. So if it's better there, it will be worse there, essentially. And there's a well-known paper on that topic from '97, basically, which proves that. So we were asking, okay, is this also true in systems? Is this also true in systems biology? Because what was considered there was a very specific class of problems. And we evaluate this on these 20 models. Each point here is a model and the corresponding data set, and we performed a fitting basically for this. And here just a small small zoom in into some of the results. What we compared here were two different optimizers, interior point versus clusters and region reflective. Nothing special, implemented in tons of toolboxes basically. And what we have here is the performance of these two methods. If it's above this line, actually trust region was better. And if it's below, it was interior point. And what you can see is a clear trend that in most cases, trust region is better actually than interior point. This kind of indicates that the no-free lunch theorem might not apply. And this can be also shown when looking at global optimization methods and so on and so forth. So some are simply better for models we find and encounter in systems biology. And the reason for this is that our model, well, our problems are not random, but they are coming from a certain class. So it really makes a huge importance to select the optimizer, and there is indeed a free lunch. And what we showed, for instance, in these papers was an evaluation of what optimization methods might be best suited for different classes of systems biology problems in terms of model size and so on and so forth. I have to apologize for the background. We are currently in quarantine and my daughter is playing. So just see that as an added benefit. I hope it doesn't distract you as much as me. The second thing we were checking actually, and usually if I would know if this would be in person, I would ask now in the audience, how do you expect actually the number of iterations of an optimizer to increase with the dimensionality of the problem? And what most persons expect is that's linear or quadratic. However, there was actually in a paper or a book already not too long ago, okay, almost 20 years by now, already the statement that at least for any method which is based on a Newton step, the number of iterations should hardly grow with the dimensionality of parameter space and the corresponding constraints, which is kind of interesting because it says independent of whether you have 5 parameters or 500, you would roughly expect the same number of iterations of any type of local optimizer. And this appears strange to many persons. So we said, okay, does this hold true? In the model collection we had already back then, we had models of quite different sizes here, ranging from really a few parameters. I think the smallest one has 4 parameters to something which had like 300 parameters. And you see here the average number of iterations of the corresponding optimizer. And what we find here is hardly any correlation, meaning that we did find that, which is obviously quite nice. So there's no dependence of a number of iterations on the problem size, meaning that our methods, at least if they are based on gradients and so on, can really nicely scale up to very complex models and potentially also to complex model classes. With computation time, it's a bit different. Usually if you have more parameters, you have more experimental conditions, so more simulations to run, and so on and so forth. So for computation time, we see a roughly quadratic dependence, meaning if a number of parameters doubles, the computational complexity increases by a factor of 4, at least if we use forward sensitivities. And that again can be changed if one goes to alternative gradient evaluation schemes, basically, which can push down with linear growth in complexity in this objective function and gradient evaluation down to something which is linear. And these are techniques which are fairly well known in our fields, which are, for instance, also employed in machine learning, basically. So their so-called backpropagation techniques are employed to reduce training complexity, and similar approaches can be also applied here in systems biology to really accelerate the training of models and to facilitate a large analysis of large scale problems. And again, this is quite positive news here, because it essentially means that we can scale up this type of mathematical and mechanistic modeling to relatively extreme scales, because essentially we have only a linear dependence in the number of parameters. And the last thing I would like to briefly mention, which holds true in our experience for various model classes, not only for ODEs, but for many others, is actually that parameter transformations can play a big role. So there was already in 2013 a statement in the parameters. For parameters that are by definition non-negative, a log scale should be used in the parameter estimation problem. And there was a lot of discussion about that forth and back, and I still see many studies in which this is not the case. So we also set out to evaluate that, just using the same optimizer, multiple ones, on linear or log transform parameters, basically. And essentially for all models we have in our benchmark collection, the log transform optimization, log transform parameters performs at least as good as linear. For most models, it's orders of magnitude better. There are different reasons for that. Here just an illustration for a very small system, X being synthesized and degraded, and we have an observable Y basically, which is proportional to X. If you look at that in the natural parameters, you have here a very strange curvy objective function landscape. If you look at this in log scale, basically everything becomes nice, and actually level sets are perfectly convex, meaning that something which has has information about the second order structure of optimization problem will jump in a single step more or less to the optimum, which is really nice to have because it decreases iterations. So this transformation plays a crucial role, and if you're not doing so yet, I would really recommend trying that out onto your problems. So that finally brings me now really to some applications where we were trying to look at really high throughput data. They are nowadays available for many different settings. Basically, I mean, obviously in the field of genomics, this is a hot topic, but also in many other fields where robotics platforms are nowadays used to automate processes and so on and so forth. So this is a project which has been going on now for 5 years with some intermediate results. And I just want some persons, if you already listened to one of my talks, might have seen parts of the story. So I just, for those of you, I will give an update. For the others, it's something new. So what my group is really interested in are signaling pathways in the context of cancer. And there was a nice paper in 2018 in Cell, which analyzed and studied which pathways, and these are basically different pathways are shown on the x-axis, are altered in which cancer types, which are shown on the y-axis. And the numbers in here, which you probably are not able to read, but which are also represented by the color coding, show how often something is altered. And what you can appreciate is that essentially for no single cancer type, there's only one pathway altered, but there are usually multiple pathways which are altered, which means that if you want to understand these cancers and potentially then also identify reasonable better treatment strategies, we shouldn't look at a single pathway, but in multiple pathways, basically. And this is what we kind of set out, which is one of key research topics in my group, to look at larger and larger and more comprehensive pathway descriptions in the context of cancer in a data-driven way. What we started developing with a company called Alacris Theranostics, which is located in Berlin, was an integrative model of cancer signaling. And what this model captures are multiple pathways. So what you are seeing here is not individual species, these are individual pathways. So there are multiple ERB-B-related pathways, there's the RAS pathway, the AKT pathway, and many other pathways which are captured. And this model is structured in 3 layers. Protein-protein interaction, as you would usually write it down. Then there is this level of gene expression profiles, where we essentially look at exome sequencing data, what is actually altered in a specific cancer compared to wild type or germline, depending on whether you have germline data available, or this genetic profile. And then we look at also the expression levels, basically, of these different components in different patients or cell lines. What I'm going to focus on today is cancer cell lines, essentially. And this is again one of these ODE models, here relatively large. Just to give you a scale, this was at least in 2018 the distribution of model sizes of curated models in the biomodels database, which is as I mentioned previously, one of the reference databases. And depending on which parameter you look at, either number of reactions or number of species, either second or third largest model basically in this repository. And by now we increased it again by a factor of 2 roughly. So we are looking here at very large scale models. Overall about 2,000 biochemical species and about 5,000 biochemical reactions essentially. And we use this model to actually analyze a published set of high throughput data. This is actually the newest release of a cell line encyclopedia, Cancer Cell Line Encyclopedia, CCLE. Really nice and helpful data set to understand signaling in cancer cell lines basically. And I've been quite a few studies also on how these cancer cell lines will relate to actual patients. The data set is truly amazing, also including, for instance, drug perturbations to more than 30 drugs, basically, which can then be used for training. Back in 2015 or 16, when we started this project, my PhD student, Fabian Fröhlich, did a rough calculation. Back then, expected computation time for training was 200,000 years. Not too helpful, and he complained about the expected length of his PhD. Nowadays, we are down to 3 days roughly on a cluster of 1,000 cores. We can also go obviously higher here, but this is only a small fraction. So there are many different things basically which contributed to this acceleration basically in training time. And this is also, in my experience, necessary to really look at this high throughput data basically. So one really has to tailor the methods such that you really squeeze out all of the small factors basically, and they are essentially adding up. So if you face such a problem, in my opinion, there are 2 different approaches one can take. So for this high throughput data, the first thing is that one really tailors it, as I just mentioned, to a respective problem class. So usually it's a problem class and not a model, and I have to be careful now with time. I will try to keep the rest relatively brief. So what you usually have here are relative measurement data. So instead of directly measuring X, it's scaled with some factor, basically. And what the standard approach is to simply define a large parameter vector and to really integrate all of these factors into this large parameter vector, and then do a simple iteration and try to optimize the full parameter vector, simulate the model, evaluate objective function gradients, check whether stopping criteria are fulfilled or so. And if not, then you continue. What we developed here, and which is applicable to a very broad class of models, and by far not restricted to ODEs or PDEs, is to actually exploit the structure here. And we defined a hierarchical approach where we really have an outer loop only the kinetic parameters which influence the simulation, and an inner loop all of these parameters which only influence the objective function, like rescalings and potentially also noise parameters. And we showed that this can substantially improve various different optimization schemes and many other things, basically. So here you see optimization runs for this large-scale model with a standard optimizer and with this hierarchical optimizer. And already after 1 iteration, we are at the same objective function level as for the normal one after dozens of iteration, basically. And that not only for 1 optimizer, but we see this consistently across many optimizers and across many models. And I will briefly skip over that. Second approach is to use methods which are successful in other fields.
Jan, sorry, I have to, please, if it's okay, just finish in a few minutes.
So 2 more minutes. So the alternative approach is, in my opinion, to use methods from other fields. I'm a big fan of machine learning, so we tried to do that. And in machine learning, there have been methods developed which actually achieve a sublinear scaling in the data set, so-called mini-batch optimization methods. I will skip over the details, but essentially what's done is to not use the full data set in each iteration, but only a subset, which results in a stochastic optimization method, but which can, for this large-scale method, result in a gigantic improvement. And we applied that again on this very large-scale model. And we essentially see a 50-fold improvement in computation times and sometimes even better objective function values, basically. And the nice thing is that these methods are also quite robust to noise. So here the performance basically not only on the training set, but also on independent test set. So regarding the prediction accuracy, so we see a very nice correlation on data which were not used to train the model, and a relatively good area under the curve of a predictor. And if we combine here different optimization results together, actually we reach up to 90% accuracy, which is quite nice. So what about more complex models? I unfortunately don't have the time to tell you, but we also did some work there basically for multi-scale approaches. But I would like to skip over that now and directly jump to a summary. So what I hope I conveyed is a kind of an overview about challenges we are facing for high content and high throughput methods. Then these hierarchical and tailor-made mini-batch optimization techniques. And what I didn't mention and couldn't describe is our work on approximate Bayesian computing and inference for multi-scale models in that setting. And there are some tools which might be helpful. And just as a concluding sentence, I think we are nowadays with this kind of, so as a community, by bringing together these different approaches, really able to look at also deep mechanistic models which capture quite a few more things than the classical modeling approaches basically, and are able to integrate this high throughput and high content data. And with that, I would like to acknowledge the collaboration partners, which were also mentioned on the individual slides, and my group, which is really amazing. With that, I would like to thank you for your attention.
Yeah, thank you very much, Jan. Thank you. We're a bit after time, but maybe we can take one quick question from the Zulip chat, which is if there's a similar result for optimizers based on genetic programming or particle swarms. Philip says, I would intuitively expect the latter's iterations to scale with the dimensionality. So
we evaluated that for this benchmark collection, also for other types of optimization methods, and we didn't encounter that the number of iteration is constant, basically. So usually we find that actually the number of iteration is increasing. Actually, quite a few of standard particle swarm methods and so on are anyhow very greedy and just don't implement proper convergence characteristic, but just kind of use up their budget, basically, and then stop after this is used up. Okay, thank you very much. Please feel free to ask more questions or also answer. There's another question in your topic now, but I would like to continue with the presentations. And the next presenter is William Lövfors. He's a PhD student in systems biology in his last year at the Department of Biomedical Engineering at Linköping University. And he will present his work on modeling of mechanisms in adipose tissue with the ambition to aid drug development for diseases like type 2 diabetes. Please, William.
Thank you very much. Thank you for the nice introduction. I will just start my screen share. There we go. Do you see my screen? Perfect.
Yes.
I would also very much thank you for inviting me for giving this talk, and I would also thank you for putting me right after John. He gave some very good introductions to the things I will be talking about, so I can just skip those at all now. Our models that I will go through have all been based on ODEs as well, and sort of the main goal of this talk is to sort of touch upon how you can combine, or how we will combine models to make a large, more complex model. So that's similar to many things that you mentioned. I think I will just start a bit briefly by talking about this title. If you have read it, it says towards a comprehensive and multi-level module for metabolic control of the adipose tissue. I guess that some of you think I might have misspelled this title and that it should have been model instead of module, but that's not actually the case. But I will leave that as a short teaser for tomorrow, because then Gunnar, my main supervisor, will present our work towards creating a multi-level digital twin. And to make this twin, you need to, of course, have separate modules where the adipose tissue is one of them. So then one might ask, why do we want to study the adipose tissue? Well, one short answer is diabetes. A slightly longer answer is that diabetes is a bad disease that you don't want. It can cause cardiac complications, broken down blood vessels, and many other things. And the problem is that diabetes, at least when I'm talking about type 2 diabetes, is ever increasing in the world. It is connected to being overweight, and the population in the world is steadily growing more and more overweight. So we really need to understand this. And the problem now is that we fully don't. And one of the reasons is that this is a complex disease, and there are many different things that sort of interplay and have different crosstalks. So I guess I will say the rhetorical question, what do you do when you have a complex system that you want to analyze? I hope many of you answer mathematical modeling. That's at least what's my answer. So just in brief, in this project, or the things I will present will be based on, let's see if this works, yes. So this is the fat cell. From the fat cell, or tissue, we can take out the fat cells and process it in vitro to get in vitro data. But of course, the fat tissue isn't working all on its own. It's a part of a much bigger picture. And we can also extract data from this bigger picture to get in vivo or in situ data. Our first attempt to create the model for the fat tissue was what I did in my master's thesis. I tried to create an automatic way to go from a small model of insulin signaling and scaling it up towards a full model for the entire fat cell using phosphoproteomics data. This here will be a short animation of the actual results. So this model was expanded iteratively. And what you're seeing here is things that were added to the model that was actually able for the model to explain the data. So the data that we had looks similar to what Jan presented earlier. I will just open my laser pointer here. We have time, often we have time on the x-axis and some response. So here the error bars are the experimental data, and the line is the model simulation. The problem was that when we tried to also explain some independent data that we did not use to train the model, we got different results. So we classified in this project 3 different outcomes. Either that the green line here, where we were within the uncertainty of the prediction data, that we were going in the right direction, shown here by the purple line, or that we were above or going in the wrong direction when we did the prediction. And unfortunately, when you look at all of the proteins that I showed earlier, the results weren't super convincing. You would like to be within these 2 quadrants, but there were a lot of simulations that ended up being incorrect. So we had to scratch that approach. We tried to make changes to the algorithm, but I never got around making good predictions. And the model that can't make predictions is not really useful. So the second attempt was to take our small mechanistic models that our group has developed over the years, where I have been involved in some of them, and sort of try to combine those instead. So this here is a cell. You can see the membrane as these black sort of lines here. On the adipose tissue, there is a few receptors. The ones that we have been working with is the insulin receptor, the alpha-adrenergic receptor, and the beta-adrenergic receptors. And these we can stimulate with different chemicals: insulin, phentolamine, epinephrine, and isoproterenol. And this will trigger things inside the cell. And then the 3 sort of main functions of the cell can be perturbed. So we can look at the glucose uptake, the breakdown of fatty acids and the release of fatty acids to the outside of the cell, and also the release of adiponectin. So I will start talking a bit shortly about our model of glucose uptake. And this work has been done iteratively over many years. I will give you sort of snapshots through this development. So the first thing that was studied was the insulin receptor here, and that was done already in 2008. So here is some data and the model simulations. You see the red sort of, it's supposed to be error bars, but I guess this is maybe a bit too narrow. But you see the red points here are the experimental data, and the blue lines are the model simulation. Here we have a time series for the insulin receptor and also a dose response with increasing doses of insulin on the x-axis. And I say that this agreement looks fairly good. They went on to study the insulin receptor protein 1, the insulin receptor substrate protein 1, IRS1. And again, the model was able to explain the data. More extensions were made, and when they went all the way down to PKB, again, the model is able to explain the data. And lastly, a feedback mechanism was added back. And in the end, they went all the way down to glucose uptake. So most of the data here is these intracellular in vitro data, but there's also one that's actually in vivo. And that was by connecting this model to the Dalaman model, which I will not present here, but I assume that Gunnar will talk about this tomorrow. The next major step was to introduce diabetes to these models. And then things change a bit. Again, I start with the insulin receptor. The dots are experimental data. The lines are model simulations. Blue represent healthy controls and red represent diabetic patients. So the model is able to explain both normal and diabetic data. This is for the insulin receptor. We of course have more data for the things I've shown already. IRS1 looks really good. I will present a lot of data now really short, so don't look into all the small details, but try to sort of get the overview of it. So we also have for PKB and the glucose uptake, and it's still able to explain the old things. Then they did some more extensions to these models. They added path through mTORC2, giving an activation on the serine 473 site. And that looks good, I would say. They found out that there was another protein along the way to the glucose uptake from PKB, AS160. Of course, we have data for that as well. And then there is another pathway that's sort of independent from this one, but it goes through MEK and ERK. And that looks convincing. I would say I'm going to say this like multiple times, so you will just have to get used to it. Then we of course have another pathway or another action. That's the activation of ALK1. And there's data. We have activation of FOXO, and there is also data. So, and then I think I have one more step actually. So we have also the activation of SXK1 and SX. So a lot of things in the model, a lot of data. So I hope you're at least convinced that this is within the ballpark of right at least. Of course, this is a simplification of reality, and it will never be the exact solution, but it's behaving reasonably good, I would say. The next thing we did, or actually what I did when I came into the picture, was to model the release of adiponectin. Adiponectin is a hormone that's released from the adipose tissue, and it's sort of has been connected to have a healthy effect towards diabetes. It's been shown to be insulin sensitizing, not desensitizing. And in essence, we have this adiponectin hormone inside vesicles inside the cell, which will migrate towards the membrane. It will be primed, and then it will be fused with the membrane, and the adiponectin will be secreted. And there is also endocytosis going on in the cell to keep the balance. If you would only have exocytosis, the cell would grow to immense sizes. So you also need to have endocytosis. And this is triggered by a few different intracellular mediators. The ones that we have looked at is cyclic AMP, ATP, and calcium. And to make things a bit more interesting, we also added a pipette straight into the cell and infused these mediators into the cell this way to get really high concentrations, or at least concentrations that could trigger this release. And through this, we could figure out that the mechanisms by which these act is that cyclic AMP triggers both exocytosis and endocytosis. This effect is augmented by ATP, as well as ATP is driving the migration from this reserve pool to the releasable pool. And calcium augments everything. And I will show you some data as well. This here is data for different combinations of these intracellular mediators in the pipette. And we can see that the model appears to explain data reasonably good. The next step was to add extracellular stimulus as well to this system. So we added adrenaline or epinephrine outside of the cell. And this triggers a cyclic AMP response or increase. And of course, we have data for that as well, which I think looks good. So the last part I would like to talk about is lipolysis, the release of the fatty acids. here we actually used all of these stimuluses that I mentioned before: insulin, phentolamine, epinephrine, isoproterenol. And we measure, amongst things, the release of fatty acids and glycerol. So you can see that one pathway is sort of shared with adiponectin release, the actions through the cyclic AMP. There is also a degradation now of cyclic AMP that we didn't model in the adiponectin release paper, which is done through the protein called PDE3B. This is influenced in 3 different ways through the insulin receptor. One goes all the way through here to PKB that activates PDE3B. One goes directly from the insulin receptor, and it's actually not activating PDB, it's inhibiting PDE3B. And one indirect way that goes through the alpha-adrenergic receptor. Cyclic AMP will activate PKA, which has a small feedback loop. And then it will trigger a cascade that breaks down the triglycerides into the free fatty acids that's diffusing or transported out of the cell with glycerol. And of course, there is a reesterification where the fatty acids can be stored again as triglycerides. And this also has diabetes, or we have implemented diabetes into this. I will show you some data now. Here are time series where we have given different combinations of these inputs. The first here is with only epinephrine in increasing doses. Maybe it's a bit hard to see, but there is 2 gray bars here, which represents an increase in adrenergic stimulus. Then we have another one where we have instead given isoproterenol, the purple one. And here we have the green one with the combination of epinephrine and phentolamine. And you can see that the model is explaining data good. We also have some dose response data for glycerol, for PKB, HSL, and the release of fatty acids. And this, I will wrap up soon. This slightly more yellow color here is actually fatty acid release from diabetic patients. So I will do some fancy animations. I have presented an insulin glucose uptake path or pathway. I have detailed another independent adiponectin release pathway, and I have also went through a lipolysis pathway. So the goal now is to combine all of these into one model, and this is unfortunately haven't been done yet, but it's what I'm supposed to do soon. Maybe we can get something published next year. That's at least what I hope. And that's everything. Thank you. Does anyone have any questions? I will also bring up some references for the works that I presented. Thank you.
Yeah, thank you very much, William. So let's see. There are no questions yet in the chat. Oh, now there was one coming up. So how did the errors, were they kept under control when adding more interactions?
Can you repeat the question?
Sorry, how were the arrows kept under control when adding more interactions?
How were the arrows kept under? I'm not sure I really follow. Are the questioner asking about the adiponectin release and the pipette?
I guess I can chime in if that's, sorry, just to yeah, no, I was curious about error accumulation. So when you extend the model with more and more interactions, I assume, I'm not familiar with the formulism, but I assume there is to each interaction some error, some
error. Okay, yeah.
Yeah.
I guess, I mean, in all of these steps, this isn't just like one model being done and then one model being done. Of course, this is done slowly and iteratively. So you try to, you start with a small model, you try to explain some of the data, then you extend the model iteratively, slowly and slowly. And hopefully if you have the correct structure, you should be able to explain all of the data with the same structure or the extended structure. But it's, I mean, through every sort of model iteration, we allow all the parameters to be re-optimized.
Okay, then there was a question how you plan to integrate the models.
Yeah, that's a good question. I haven't really planned that much. I think I will start with trying to interconnect the glucose uptake part with the lipolysis part, and then try to add adiponectin in a second step. I haven't thought about any software or specific routes, but there is a lot of touching points. So hopefully it will be possible.
Great. And then Nick seems to have raised his hands.
Yes, hello, William.
Hi.
Normally, I actually don't ask this type of questions, but it's because so the AMPK part, which I feel very passionate about because I do most of my research on its yeast homologue, is missing from your model. And this is a bit quite surprising to me since it's shown that the pathway is driven by ATP. It is heavily involved with the fatty acid breakdown and also with the GLUT4 expression. So I was wondering, is it you just didn't stumble upon it, or you stumbled upon it and found that it has no importance in the data?
No, I don't think we actually have any data for that. That's probably one of the main things. To be honest, I have mostly worked with the hypothesis and the data that we have been given by our collaborators. But I should definitely look into AMPK more. I've heard of it multiple times and seen it in multiple places, so I know it's something that's relevant. But yeah, we should really look into that a bit more.
Because I would expect since you're in your model your pKa and the cyclic AMP was quite. I guess it's black boxed together in that component.
Yeah, I mean, this is a sort of minimal model, so a lot of different sub-steps have been simplified. But we should definitely look into this a bit more.
Yeah, because it's actually quite heavily involved in the GLUT4 expression and then also bringing it to the cell membrane.
But it's mostly active during low energy status, if I understand things correctly, like hyperlatent.
Yes.
Yeah.
Yeah.
And we haven't worked with that so much yet, but we are going to now. So it's a good comment.
Okay, great. Thank you very much, William.
Thank you.
Then I think we can continue with the next presentation, and that is Jan Zrimec. He is a PhD in bioinformatics and is now part of the Selesniak lab at the Department of Biology and Biological Engineering at Chalmers. From LinkedIn, I found that he describes his work as AI plus data science in brackets times regulatory genomics plus systems biology in brackets. We're happy to have you. Please, Jan.
Okay, hi everybody. I'm just starting to share my screen. Okay, so you can see it, yeah? So, hi, I'm Jan. I'm from the group of Alexei Selesniak here at Chalmers at the division SysBio. And I'll present the main project I've been working on since I came here in 2018. And this is regarding deep learning. So applying deep learning to learn the regulatory grammar in DNA. And trying then to use these models to help with protein expression engineering. So why is this important? Just a second. Seem to have a, okay. Yeah. So why is this important? So understanding the regulatory grammar of gene expression can help us to fine-tune how cells function. And this is important for solving problems in both medicine and biotechnology. In the process of expression of DNA to RNA to protein, multiple studies have shown that there is a strong agreement between protein abundance levels and also mRNA levels. So this suggests that transcription is actually a major determinant of protein abundance. So in our study, we actually just focused on transcription. So we obtained a huge data set of practically all available RNA sequencing experiments at that time, which covered also the majority of experimental conditions. And this data is plotted in this figure. On the x-axis, we have the transcripts per million, log ppm. And on the y-axis, the protein coding genes, which are sorted according to their median expression level. So what we see here is that the variation of gene expression per each gene is actually much smaller than the whole dynamic range of expression levels that we have in our data set, which shows that actually these expression levels per gene are quite well conserved across all the different conditions, meaning that genes that are highly expressed in one condition will likely be highly expressed in others, as well as vice versa for low ones. But this also suggests that actually this phenomenon is likely encoded, at least these basal average gene expression levels are likely encoded in the DNA sequence itself. So I want to talk a little bit more then about what this means, how this is encoded in the DNA sequence. So here we have a gene structure, or as we define it, a gene regulatory structure. So the CD has the coding regions, so the coding sequence of the gene, and also then different regulatory regions, which are promoter, untranslated regions, as well as terminator region. And all of these are actually involved in the three different steps of the process of transcription, which are initiation, elongation, and termination. And of course, it's well known that these regions carry a lot of regulatory signals, so DNA sequence motifs, for instance, transcription factor binding sites in promoters, Cossack sequences in 5' untranslated regions, as well as some additional 3' processing elements actually related to termination of transcription. And on top of this, we have the codon usage, so codon optimality, so different codon usage with different genes, as well as the nucleosome positioning code, which is actually important across all of these regions as it defines the accessibility of the DNA for enzymatic processing, especially the RNA polymerase. So we see that there is actually a rich regulatory grammar that we would need to model in order to be able to test if we can predict these expression levels merely from the DNA sequence. So luckily, in the past years, there's been a lot of development of these deep neural networks. And here we have a motivational example. So a very complex image that a stack of two different types of neural networks is able to, first of all, decipher, so decode what is going on in the image, and then translate this into human understandable language. And these same networks, which are convolutional networks and recurrent networks, can also be used for DNA sequence analysis. So the way convolutional networks work is they scan a receptive field and actually learn different motifs which are important or which make up this image, or in our case, the DNA sequence. And recurrent networks can then learn the sequential information in the DNA, such as different positioning of motifs and such things. But the problem is with recurrent networks that they're very hard to train compared to convolutional ones. And actually with certain tweaks, convolutional networks can achieve similar results as recurrent networks. So in the end, we just used convolutional stacks of convolutional networks, so three convolutional networks with two dense layers on top. So we were training deep models to predict these target variables, which were the median expression levels. And we used as input data, of course, this as much as we could of this gene regulatory structure that we defined, which included over 2,000 base pairs of DNA sequence. So this was defined actually by testing with shorter sequence sizes of these regions. And we found that this gave the optimal results, but we were also limited a bit by the size of the model. So this was a good trade-off. And we also then used the codon frequencies from the coding regions, as well as variables related to mRNA stability. So from what is known in literature, all of the important information for predicting gene expression levels. And of course, first we built models with data from yeast. So this figure is also based on data for yeast, for Saccharomyces cerevisiae. And with this, we obtained models that could explain over 82% of the variation of gene expression levels in the testing data set. So we wanted to test this further with different model organisms. So we then built models and performed all this data parsing and model training with model organisms spanning from bacteria to human. So a wide range of different genomic complexities as well as regulatory complexities. And we see that on average, 60% of the gene expression levels are, so 60% of the variation of gene expression levels actually explained by these models across all of these different model organisms of varying complexities. So we then further tested the yeast model. First of all, comparing it to shallow, so comparing deep modeling approach to shallow classical machine learning models. And the important thing we found is that the main improvement of the deep learning approach is actually that it can extract the information from the DNA sequence itself. So it can learn the important representations of the data, so the motifs, the regulatory grammar by itself. Whereas with shallow models, we need to actually give them a certain representation of the data, and usually camera frequencies are used for this. And this has inherent limitations. We then next tested how different regulatory regions or combination of these actually affect the prediction of expression levels. And we found that each region actually carries, so that all regions actually jointly contribute to prediction of expression levels, and each region carries a certain amount of overlapping as well as a certain amount of new orthogonal information relative to the other regions. And then we tested this a bit further by analyzing the mutation rates across 14 orthologous yeast species. So they're orthologous genes, 14 related yeast species. And we found that, so we measured a medium level of correlation, which gave quite strong evidence actually, that there is, so that these regulatory regions, especially promoters and terminators, co-evolve with their coding regions. So what this shows is that this gene regulatory structure is actually not only interacting across the different regions, but is actually a co-evolving unit. So next we wanted to test what is the actual regulatory grammar. So what are these models learning? What type of regulatory grammar are they extracting from the DNA sequence? And for this, we used a quite pretty straightforward approach, which was actually to occlude with sliding windows all the input data, and then measure the perturbations this has on the predictions of the output of the models. And we measured thus a relevance score, which showed the importance of each of these small occlusions for the model. And from this relevance profiles, we could then mine and actually reconstruct the DNA sequence motifs, so the regulatory grammar. And we found that indeed, this reconstructed motifs actually cover a large amount of the known yeast regulatory grammar, spanning all these transcription factor binding sites and additional motifs across all of these different regions, showing that actually the models seem to learn all of this, plus some additional new grammar. And so we wanted then to analyze this a bit further. So we used association rule mining, which is used in market basket research, market basket analysis, and where we treated, so each gene was actually treated as a basket of motifs, and we wanted to extract the motif co-occurrences across the genes. And here we see, for instance, a plot of the amounts of different co-occurring motifs, and we see that there is a large, so over 8-fold increase in the amount of co-occurring motifs across all the different regions than in any single region alone. So they tend to co-occur across all this whole gene regulatory structure. And here is an example with 4 genes that share a common transcription factor binding site, so an NHP6B marked blue. And they carry then additional motifs marked red, which with which this transcription factor binding site co-occurs, which actually these red motifs define or define the expression levels with this transcription factor binding site over a large range of different expression levels, practically 3 orders of magnitude. So we then asked, okay, so from the whole range of expression levels in our data over 4 orders of magnitude, how much we can actually retrieve by analyzing motifs or these motif co-occurrence rules. And we see that whereas with motifs, we can extract 57% of the retrieve, so we can retrieve 57% of this whole range of expression levels. With motif co-occurrences, we can retrieve actually 84%. So Basically, the motive concurrence rules seem to be the most important part of this regulatory grammar of yeast, and they define practically the whole range of expression levels. So finally, we then asked if we can use these models of the learned regulatory grammar to somehow guide or help with gene expression engineering. So first of all, to consider how people actually usually approach this is when they want to fine-tune the expression levels of certain gene, they just usually focus on a certain region like the promoter, and they swap it out with a known weak or strong variant without considering any of the other regions which we showed can have quite a large effect, these interaction effects. So we tested this computationally. Of course, we had the models. So we so we just kept the promoters fixed with the different genes, and then varied the terminator regions with all the different variants in our data set, which gave 18 million combinations, so 18 million different constructs to test. And what we find is that per each gene, there is actually a large amount of regulatory freedom over 2 orders of magnitude around its native expression levels, which can be unlocked just by swapping out these different terminator regions. And so promoters, for instance, that are weak can be made to be stronger, and vice versa, strong ones can be made to be weaker. And also when we kept the terminators fixed and then varied the different, so with different promoters, we obtained very similar results. So we wanted to test this experimentally, and we designed new constructs. Here we had to replace the genes with GFP, of course, to measure the fluorescence. So we chose native promoters that spanned relatively, so they span from relatively low to relatively higher expression levels, so a certain range. And then also chose, based on predictions, terminators that were strong or weak, so that could either upregulate or downregulate the expression levels of these promoters, so these constructs. And we see that we achieved actually quite good correlation between predictions and actual measured fluorescence intensities, so correlation of approximately 0.65. And here on this image, we see the relative change then of the gene expression by replacing terminators with the weak and strong terminators relative to the native ones. So just the percent change. And on average, we see that we can drive expression levels approximately 20% up or down by swapping out the terminators, as well as we can achieve certain constructs that have a much larger change. So it's good also for finding some completely new combinations. So what this shows is that we can learn, we can build models of this regulatory grammar just by learning from the native genomes. And this can be used then to guide gene expression engineering without the requirement for any synthetic approaches, which can be costly and time consuming. So that's it from me. This study is actually has been available in bioRxiv for quite some time, but we're quite excited. It is in principle accepted in Nature Communications, so we hope it will also be accepted and published soon. Let's say by the end of the year. So if you're interested, stay tuned. And it was a collaborative study across multiple departments. A lot of these people are co-authors on the study, but I'm also thanking certain people that are helping now further develop these methods and especially test them in the lab. And first and foremost, I want to thank Alexei for giving me the opportunity to work on this very interesting research. And also with that, I want to thank the organizers of the conference for giving me the opportunity to present this work. And thank you, the audience, for your attention.
Yeah, thank you very much, Jan. We have one question from the previous Jan, who is interested in if there's an intuitive explanation why the terminator region matters so much.
Intuitive explanation why the terminator region matters so much. So it carries certain signals related to termination of gene expression, but also there is, we found, just let me show here. So we found, of course, a lot of overlap with this known regulatory grammar, but there's also certain signals. So a lot of new signals actually across all of these regions, which well, which are new, so, or which did not overlap with the non-regulatory grammar. So there might be new signals related to some different processes, to some unknown things that might, that seem to carry a large importance for at least prediction of gene expression. And as we showed also with experiments also related to what's happening in vivo. So yeah, there is importance of these regions related just to what is known about the process of transcription, as well as to carrying potentially some new things that still need to be analyzed further, or also are known, but we just did not include all of this information in this paper or in this study. So I might have easily skipped also a lot of this additional regulatory grammar related then to post-transcriptional processing, or maybe additional stability of mRNA and things like that, that might actually also affect. So it's a very complex, it's a complex system that we, with this study, try to, because we try to explain what's happening, try to simplify it to a level that that can actually be managed to explain in the study. And we also with this, still the study is huge, and the paper, for instance, is very long, trying to explain all of this. But of course, likely we also left out a lot of things. Plus, there might be some things that are unknown. So I hope that answers the question, or gives some pointers to what could be the answer.
Okay, are there any more questions? If not, we can thank all the speakers, the first 3 already once, and we have a little break of 5 minutes, and then at 14:30 we can continue with the next speakers. Thank you very much. Okay, so welcome back, everyone. I hope most of the people are back from the short leg stretching break. I think we can directly start with our next speaker. That is Erik Bülow. He is a freshly graduated PhD from the Department of Orthopedics at Gothenburg University. So big congratulations on your PhD. And he works with statistical models applied in a more clinical setting. Thank you for coming, and please, Erik.
Thank you very much. I'm very happy to be able to speak today. I hope you can hear me. So let's share my screen, which I hope you can see now. So this is about orthopedics, and more specifically, it's about hip prosthesis or hip arthroplasty. So most of us here today are quite young, but it's kind of likely that when we get older, we will need a piece like this. It's a metal piece that you put in your hip. So I have illustrated this talk like, let's see, like this. So you might have something called hip osteoarthritis, which is a degenerative disease affecting your hip. It might hurt, and it might be quite difficult to walk sometimes. Although it's not really that dangerous, you can survive with it. It doesn't really affect your overall survival or anything. But you might think that, okay, you can try to treat this with physiotherapy, physical training, etc. But in the end, you might need to replace your hip with an artificial joint. But it's a decision to be made. Is it worth the risk to undergo this surgical procedure, which is quite extensive, actually? So you might imagine yourself to put a hip, an artificial hip joint in your bone here, but you want to look into the future. Will I be able to dance again and to move freely, or is the risk to die actually substantial? This is a very common procedure worldwide, but also in Sweden. So if we look at the incidence of inserted hips from the late 90s to some years ago, we see that the numbers of elective hip procedures has almost doubled. It's the blue line here. Most patients are a little bit younger than 70 years old, and 60% are females. There are other reasons as well why you might need a hip prosthesis. For example, a femoral neck fracture, meaning that you break your bone. So you need an acute operation. those are the other lines further below. Those patients are usually older, over 80 years in most cases, and 75% of them are female. But we will here focus on the elective cases with osteoarthritis. So I work for the Swedish Hip Arthroplasty Register, or as it's now a part of the Swedish National Joint Register, since it has merged with the Swedish Knee Arthroplasty Register recently. So we collect data since 1979 on all procedures performed in Swedish hospitals, both private and public. We have personal identity numbers, and we looked at all the patients in our database. We sent a list of personal identity numbers to the Swedish Board of Health and Welfare. They have additional register for all patient visits in hospitals in the whole country. And at each hospital visit, if you have any diagnosis, they are recorded into this register by the International Classification of Diseases, version 10 since 20 years or so. And also if you get any medical prescriptions, those are recorded in a medical prescription register. The data was sent back to us, but now without any personal IDs. And simultaneously, this list of personal identity numbers was also sent to Statistics Sweden, who found data on educational levels, civil status, income, etc., and send this data back to us. So the reason that we want to use this sort of data is that we want the patients themselves to be able to estimate their probability or their risk to die within a certain period. So we cannot really use any more advanced molecular data or something. So all of those data should be known by the patients themselves before they decide to take on this surgery. It's quite a large data set. We included quite many years, and we have data for the whole country. Actually, I only had access to a normal laptop computer, so we had to think about quite carefully how to program these things. And there are a lot of diagnostic codes as well. In the Swedish version of the International Classification of Diseases that we use for those diagnostics codes that are recorded at each hospital visit, there are like 20,000 different codes or something. So usually, in order to use those in practice, you combine those into broader categories like heart failure or diabetes or HIV or cancer or whatever. And two common ways described in the literature are called Karlsson or Elixauer comorbidities. Those codes are not really recognized in alphanumeric order either, so we had to code those using regular expressions. But we implemented this in a R package that is actually also available online now, not yet on Cron, but hopefully soon. And it can be used with an MIT license. So when we did calculate Karlsson and Elixauer comorbidity, we tried to use those to predict mortality or survival after the surgery, but we found out that that wasn't possible, neither for patients with hip osteoarthritis nor for patients with an femoral neck fracture. We still thought, however, that comorbidity is probably quite important to predict the remaining lifetime of a patient, but probably not using those standardized comorbidity indices. So instead, we wanted to study our patient cohort in more detail to find out which comorbidities were the most important ones for our patients. So using the data previously described, we ended up with 53,000 patients from Sweden. Actually, only 175 of those died within 90 days. So this is a very good surgery. Almost everyone will survive. As I said before, if you have hip osteoarthritis, you're otherwise healthy. You're actually more healthy than the average population, because otherwise you wouldn't even consider to get this surgery to begin with. This is good, but it also makes it a little bit more difficult to predict which one who will, unfortunately, not survived those 90 days. After we developed the model, we also wanted to evaluate it using external validation data from England and Wales. And we included 125,000 patients from there. And we had a little bit higher proportion of deaths there, 0.5%, 625 patients. But we have a lot of patients. We have not that many deaths. We have quite a lot of data. So we wanted to use some technique to select the most important variables to use in a model. Also, the model itself in the end should be very easy to use. So we should not include that many variables. As I said before, the patient themselves should be able to use this model. So we had all the data in the derivation cohort. Let's see here if I can get the, yes, here. We took bootstrap samples, 100 of them here. For each of those bootstrap samples, we, let's see if I can, yes, here. We took an additional 100 bootstrap samples again. And for each of those, we applied a logistic lasso regression with a lambda penalty term found by cross-validation. We estimated the coefficients. Some of them were shrunk to 0. Some of them were not. We took their absolute values as a measure of variable importance. Not a very good one, I must admit. Some variables were correlated, but just to get an overview which variables had some influence on the outcome. We averaged those numbers. We put the coefficients into a representation like this. We used the piecewise linear regression to find out which coefficients were the most important ones. We took a list of those coefficients. From the 100 bootstrap samples, we tabulated those and found which variables were selected many times. All potential predictors that were selected at least once were then included in a logistic regression model. Also, a reduced model was made where we only included variables that were selected at least 1 out of 3 times, so 33 times out of 100. And then we used those variables in a simple logistic regression model. Now I have some, let's see, yes. So we computed sensitivity, specificity. We made the receiver operating characteristics curves, and we estimated the area under those curves as an AUC value. So this value, as you probably know, is a value between 0.5 and 1. The higher, the better, indicating discriminatory ability of the model. Traditionally, in our field, values above 0.7 are considered good. And for the main model and for the reduced model, we had values around 0.8. They were very similar, so we choose to continue with the reduced model since it's just simpler. We evaluated that model using the British data from England and Wales. The AUC number decreased slightly, but not with statistical significance, and it's still above 0.7, our magical, but of course subjective threshold. This can be compared with more traditional models, including, for example, Charlson and Elixhauser, with values below 0.7. And this final model includes something called ASA class, which is an assessment of the patient's overall health prior to surgery. And it's also important if the patient has kidney disease, some disease affecting the central nervous system, cancer, obesity, sex, and age is also important. Not that surprisingly. The area under the curve is a measure of discriminatory ability, but we also checked the calibration, comparing the predicted probabilities to the observed proportions. And ideally, those would follow a straight line here, and they kind of do, which is good. But we cannot really use the model for estimates of probabilities higher than 5% because we don't really observe those such patients. So that would lead to extrapolation. And in the end, to be able to use this model for the patients themselves, we implemented a shiny application online. It's implemented in Swedish and in English, so the patients can go there, fill in their data. It's not much of it, so it shouldn't take that much time. And they will get an estimate of their probability, which is almost always very, very low. Because as I said, this is a very safe procedure. So it's more, I mean, you could think about this as a decision tool in order to make a decision whether to operate or not. But in most cases, I guess that patients would try it out and see that, okay, it's very safe for me, even though I might have some comorbidities or if I'm old, etc. So this project involved a lot of different sources from where we took data. But I am employed by Västra Götaland region and a department there called Center for Registers. And I also work part-time by the Regional Cancer Center. And I'm affiliated with the University of Gothenburg. Thank you.
Thank you. Thank you very much, Erik. We have one question already that is, I was wondering if this tool is in active use and advocated by hospitals or doctors, or is it more a proof of concept?
Actually, I got a message today that the article where we describe it was accepted by a journal. So now we plan to release it officially. The address that was here is just a temporary URL. It's accessible, but it's not really advertised. But very soon we will integrate this into the quality register's website. So it's a quality register, it's called. It's like a semi-independent organization. It's not part of any individual region or county, but it's supported by the SKR, Swedish Council for Communities and Regions. So it's kind of official, it will be.
But the goal was that the patient basically uses it themselves, yeah.
Yeah, exactly. And I mean, mortality, as I said, is a very unlikely outcome, but we will continue to build upon this to also try to predict other adverse events that patients might experience with higher probabilities. For example, the risk of infection, which is a very severe complication, or the risk of that the prosthesis will not work for some other reason, for example.
Would that be applicable also to other diseases at some point, like the whole setup, or is it now very specialized on this?
I don't think you can use it directly. You might adapt it a little bit, but it might be interesting, for example, also for patients with the fractures. They don't really have the same possibilities to choose whether they should operate or not, because it's a fracture, so you need to do something. But still, it might be interesting to have some estimates of their probabilities, which is much higher. Those patients are really sick compared to the patients with osteoarthritis.
Okay. Any more questions?
I was wondering about one thing. We can see it a little bit in the screenshot here, how you try to communicate this uncertainty, because if a patient gets a number like 0.06%, they might not necessarily have any understanding what that actually means. Is this like dot plot that you have there, is that the way how you communicate the uncertainty, or do you have other ways of communicating the uncertainty to the patient?
No, unfortunately, we don't really have that. I mean, in the end, we ended up with a logistic regression model. I mean, we could estimate coefficient, confidence intervals and stuff, but it would underestimate the uncertainty because we don't really take into account the variable selection process and everything else there. It's possible to do that with the delta method, I've heard, but I have not really experienced it myself. And I mean, to make prediction intervals is not really that easy either, because the observed event is either death or not, while the estimated thing is the probability. But those dots down here, we just show that patients like you, it will take 1,800 patients like you, then we can suspect that maybe one of them will die within 90 days.
Okay. I think I meant mostly the communication of the uncertainty, but we can take that maybe in the chat, because I think the time is over now.
Yeah, so yeah, please feel free to ask questions afterwards as well in the chat, also to the talks before, so don't be shy. But good, then we can continue with our next talk. That is Linnea Österberg. She's a PhD student in systems biology at the Department of Biology and Biological Engineering at Chalmers as part of the Swedish lab. And she will present a hybrid model that she used to investigate the interplay between nutrient signaling and metabolism. Please, Linnea.
So, I hope you see the correct screen now.
I think it's your presenter's mode that we see now.
Is this the correct
Yes,
one?
perfect.
Yes. Well, yes. So thank you for the presentation. Yes, my name is Linnea Österberg. And I work with glucose signaling that I'm mostly interested in. I work with the SNF1 pathway, which in yeast, which is the homologue to the AMPK pathway in human cells. And my goal is to evaluate the dynamics of the pathway and look at interconnectivity between other pathways and metabolism. So my main interest is that we have when we sense different conditions in the environment, we have a lot of different inputs for the cells. So and then when the cell senses two different inputs, they might have different responses that are in contrast to each other. So this could lead to very weird reactions in the cell unless they were coordinated. So that's why we see that in cells we have highly coordinated signaling systems with a lot of crosstalks in between the pathways. And therefore it is interesting to look at them as not as a unit, but together. But to complicate things, a lot of the molecules that the cells are sensing are also working as metabolites in the metabolism. So therefore, the responses are often very connected with the metabolic state of the cell. And therefore, it's also interesting to look at the signaling pathways in the context of the metabolism. And that is what we're interested in. And I work with the glucose signaling. And normally, or signaling pathways, you can model both using ODE models or Boolean modeling. But if you use ODE models, you often need experimental data. But that is sometimes hard to find in signaling networks with the kinetic parameters that we don't know of and mechanisms that are still a mystery. And therefore, sometimes it's easier to model them using Boolean networks where you only need to know the topology of the network. And that works by setting up different logical statements for each node, which is a component in our signaling system. And then we can use this to produce a logical steady state and then see how the dynamics looks like, but also the interactions between the different nodes. But in signaling networks, we also have complex interactions that can be hard to model using Boolean modeling. So for this, we have, my group developed a vector-based formalism for Boolean modeling, where we can take account, for example, localization, phosphorylation, we can have a DNA binding or and also if they're present or not, so we can make deletion strains in the Boolean model. So we have previously developed this Boolean modeling framework where we have this vector, and then we can produce logical statement for each part of the vector, and which makes it easier to visualize how the networks looks like dynamically, but also allows for more complex interactions. We also want to look at the metabolism, and that is very well described using the FBA modeling framework, where you can utilize the mass balance of the metabolism, and then also run the model to where you have a steady state assumption. So the problem with the FBA modeling framework is that you often have a very large solution space, and for a given objective function. And it is hard to tell which one of these solutions are the correct one. So to counteract that problem, we can make assumptions, and we can also use experimental data to shrink the solution space. So what we have done is to use enzyme-constrained modeling, where we can measure the protein content of a cell, and then we can integrate kinetic data of the enzymes that we have in our metabolic network, and then we can calculate how much of the enzyme do we need to carry a certain flux through a reaction. And since we have a limited amount of proteins, we can then limit the solution space to only biological feasible solutions. And then we also make the assumptions that we want to minimize all the fluxes to waste less energy. And then we get an optimal solution. So this is what we use for enzyme constraint model. So to look at the signaling pathways together with the metabolism, we developed a hybrid model where we use the Boolean model to represent the signaling pathways. And in this case, we're interested in the SNP1 pathway, but in order to integrate all the crosstalk and to look how that affects the cell, we also have the PKA pathway and the TOR pathway, which is shown to have a lot of crosstalk with the SNP1 pathway. And this we connect to the enzyme constraint model of the metabolism through a gene regulatory network. And then we use the results from the enzyme constraint model to feed back into the nutrient model. And this way we can model batch experiments, we can model a chemostat experiment and look at how that connects to real life data. So the Boolean module that we use for the nutrient signaling, we constructed with known crosstalk between the reactions, and we validated it against literature by comparing also the logic steady states expression profiles of different target genes of the pathways. And this we then feed into the gene regulatory network, where we get a Boolean statement if the target genes are upregulated or downregulated. And this we then connect back into the enzyme constraint model of the central carbon metabolism using by calculating the solution space for the enzymes by making an enzyme usage variability analysis. And then we have a regulation factor where we work on the lower bounds for the upregulation and the upper bounds for the downregulation, and constrain the solution space for the enzymes, depending on if they're upregulated or downregulated. Then we run the enzyme constraint model with these new constraints and then connect that back to the nutrient model using the glucose uptake rate for on and off in the Boolean statement for the glucose. And this we tested with this new hybrid model. We tested the prediction power for individual proteins with and without regulation. So we compared this to just the purely enzyme constraint model. And we found that it hugely improves the prediction. We can see here on the box plots of the error. And we can see the most dramatic change in the ability to use different isoenzymes. So here you can see an example in fermentation for the hexokinase reaction. We wanted to look at how the regulation affects pathways and which pathways are most affected by regulation. So we did this both in respiration and fermentation. And here you can see that regulation increases the exchange fluxes of glucose, oxygen, and acetate. We have an overall increase flux through the pentose phosphate pathway. And we have also a lot of change in fluxes through the around the pyruvate branching points. And this leads to an overall increase of the TCA cycle. But what's most interesting is that we can see an induced use of NDE instead of NDI, instead of only NDI. And that is very interesting because that's been previously associated with chronological aging. And by using this hybrid framework, we can then, by making deletion strains in the Boolean module, we can then point this to the SNF1 pathway and see that this is what causes this, this induced use of NDE. And you have seen that if we increase the expression of NDI, but not if we increase the expression of NDE, we induce apoptosis in yeast cells. So we also have we introduce futal fluxes by introducing regulation, and we can see it in the galactose metabolism, in the glycolysis, TCA, and in the ADH reaction, but also in the pentose phosphate pathway. So to look at closer at this, we calculated the flux control coefficients, and we could see that the highest control is located in the oxidative phosphorylation pathway. But if you compare the flux control coefficients with the purely enzyme-constrained model, you can see that by adding regulation, we have a dramatic change in the control that the hexokinase reaction has on the glucose uptake rate. And this is also something we can see in fermentation. In fermentation, the highest flux control coefficients are concentrated in the TCA cycle. And we also have futal fluxes that are introduced, but they are also smaller in magnitude and also fewer than in respiration. And we can also see an increased flux through the glycolysis and downregulation of oxidative phosphorylation pathway. And that is consistent with the long-term Crabtree effect in yeast that has been also attributed to glucose repression. So this tells us that we have the lower futal fluxes in the fermentation, but we also have higher changes of bigger changes in magnitude in the respiration case. It tells us that in the respiration we have more focus on control and robustness, while in fermentation the enzyme utilization has a bigger control of the protein allocation. And that's, I'm open for questions now. I want to thank all the collaborators I had on this paper, and also all of you for listening. And you can also look at the paper if you are interested more in details on how this type of modeling is done and how you can apply it to your research.
Yeah, thank you very much, Linnea. There are no questions yet, but I can maybe start with a small question. how have you checked what the crosstalk in the Boolean part, of what effect it has on the rest of the simulations?
No, that is not something we have done. We have not specifically the crosstalks, but we've been looking at if we delete different components in the different pathways, how that affects the flux.
Okay, thank you. Then there is Nick who raised his hand.
Hey, Linnea. I think it was slide 7 and 8 or 8. I had a question about. Yes, this one here. So with your Boolean models, the nutrients feed into the signaling pathways, but there's only the glucose uptake rate is given, while the TOR pathway's main sensor duty is for nitrogen. How do you treat nitrogen then in your model?
In our model, we have the simulations we make are comparable to the glucose limited chemostats, where we keep the nitrogen constant. But it's possible also to feed, to make a nitrogen connection as well. It's just a matter of what you're interested in simulating and what data you want to compare the model to. I don't know if that was the question.
Yeah, so your TOR pathway is constantly sensing nitrogen, but then it gets,
exactly.
It gets influenced lower down by the I get some information and send some information to the other pathways. But does it then change a lot in your model, since it only sends out information to the SNF pathway and the PKA pathway? Like we have nitrogen, that's basically what it sends out there. So there's not much change then in the TOR pathway itself when you run it.
No, exactly. The effect that the TOR pathway have is mostly by crosstalk with the glucose sensing pathways. So, and that we can also see it's quite robust in low glucose conditions and respiration, but in fermentation we have quite significant effects by the TOR pathway.
Okay, that was what I wanted to know. Thank you.
I also had a question. As a co-host, I can't seem to raise my hand, so I just started talking. So very interesting. Thank you for the presentation. I remembered that one nice thing about symbolic or Boolean or symbolic models was that in principle you could use them for querying, not only forward simulation. So for instance, can the state be in this state? So you would describe the state and then you would basically use the logical formalism to sort of retrieve a possible explanation or a possible way of reaching that state. So I'm very curious, now that you have a hybrid model, does that sort of line of reasoning came up where could you do something like that, would you think?
Yes, I think it's a lot more computationally heavy. I'm not a model. My background is in biology, but I don't see a problem with. I don't see why the hybrid model should pose a problem. It would just take a long time to make those simulations.
Okay, interesting. Well, we do have stronger computers now. Well, and getting stronger, so that might. Well, thank you. Interesting.
Any more questions? If not, of course, again, the chat will be open all the time. And I think then we can thank now Erik and Linnea again for the 2 talks. We will have a short break again now, and then at 15:20 we will start with the poster sessions. So for those who are poster presenters, I guess you know what to do, just be online in the room. And for those visiting, you will find all the abstracts, and also the posters are uploaded in the posters stream. So you can go there and check what you're interested in and then join the room. And the link you should also find there. Yes, this is supposed to take until, let me just check, until 16:20 approximately. Of course, if you are engaged in a discussion, there's no need to stop. Until 16:30, where we would ask you to be back in this main room, because then we have the last speaker for today, and that's Lars Rönnegård. Good. I don't know if I forgot anything, Philip or Felix? No?
No, I think that's everything. Just a quick reminder as well that there's tech support. So if you have any issues with any of the tools, either Zulip, or you can contact us directly on email if, you know. Yeah.
Good, great. Then see you later and have a nice poster session. I hope you had a nice poster session, and we apologize for the inconveniences in the beginning. It was a bit of chaos, but I think in the end, or very quickly, it worked, thanks to your help as well. So thank you for that. Now we come to our last speaker for today. That's Lars Rönnegård. He is a professor in statistics at Dalarna University, but he's still tightly connected to Uppsala as well. His ambition is to find simple statistical solutions to complex problems in biology. And today he will present work he did in the context of dairy cattle. So thank you, Lars, for being here, and please.
Okay, thank you for inviting me. Yes, so I'm a professor in statistics at Dalarna University, but also a researcher at SLU, the Agricultural University in Uppsala. And today I will talk about cows. So to get you in the right mood, I want to think as a cow. So typically a barn with dairy cows in Sweden has around 100 cows or so. And imagine you're a cow inside this barn. The barn is pretty big and cozy, but try to think about what in the surrounding environment is important for you and your well-being. So what could there be? And if anyone would prefer to answer, I would be very happy. So what in the environment could be important for you and your well-being?
Maybe other cows.
Perhaps other cows.
Grass.
And grass, something to eat. That's important as well. Exactly. But definitely other cows, because I mean, if you think about it and look through the eyes of the cow and think what you see inside this barn, it's lots of other cows. So there are cows really everywhere. So the social interactions that you have with the other cows are important. And that is what I'm going to talk about today and what my research project is all about. So it's social interactions in dairy cows, and it's about the interactions and the implications of social interactions for the cows. So what the surrounding cows have as an implication for the cow, really. So this is a rather large project. It's financed by Formas and the Bayer Foundation, and we got about 20 million Swedish crowns for 3 years to work on this, starting this year. So it's cow social interactions and disease transmission that we are working on. CSI-DT for short. So it's a rather large interdisciplinary project, and it's organized into 4 main parts, where different institutions are responsible for the different parts. So that's how we have chosen to organize ourselves. I'm in charge of the genetics and computations part, whereas University of Copenhagen is responsible for the disease transmission. And then we have VEXA, which is a dairy farm. It's a dairy farmer-owned organization with about 6,000 members. And VEXA, they give all kinds of advice to the farmers, including breeding value evaluation. And in breeding value evaluation, the animals are ranked according to the genetic potential. So they are ranked depending on how much milk they produce, but also how healthy they are. And then they do breeding value evaluation based on that. And the fourth partner is Rice Institute. It's one of the largest research institutes in Sweden, and they are responsible for the study of behavior and social interactions. And the goals we have of the research project, it's to develop tools for summarizing animal movement and social interactions in dairy farms. So that's kind of the basis for everything. And then we also wanted to develop decision support tools for minimizing disease transmission within the dairy farm. And we're focusing on mastitis, which is when you get inflammation in the udder. So it's a quite common disease, which we're focusing on. So based on the knowledge gained from the social interaction studied in the first goal, we look at disease transmission in the second goal. And we also want to develop methodology for breeding on indirect genetic effects. So social interactions can affect milk production. So suppose that the effect of a cow has on other cows is inherited in some way. Then it should be possible to breed for these kind of genetic effects, the social interactions effects. And in the literature, these are referred to as indirect genetic effects. So I will not go into these genetic effects and the theory behind that in this talk, but it's really interesting and has important implications on welfare. And if you want to know more on this, Peter Beimer has written quite a lot, and he's one of the collaborators on the project as well. And a good starting point is his 2007 paper. So social interactions can have effects on the other cows, and if it's inherited, then it's possible to breed on these effects as well. But I will leave that goal and we'll focus mainly on social interactions and disease transmission. The data that we're collecting, it's from 2 farms, one Swedish and one Dutch. And there's about 200 cows on each farm, and we get positioning data for all the cows every second. So every second for all the 200 cows on each farm, we're getting their position. And we also get some cow traits, like how many calves they have had previously, the disease history, and how much milk they're producing. And on the Swedish farm, what we've been doing this summer is that we've been collecting milk samples from all the cows, and also environmental samples collected around the barn. And we're doing pathogen strain typing, so looking at what kind of bacteria we have in the milk samples and in different parts of the barn. And then we're going to connect that to how often the cows meet and who they meet, to see how the bacteria is transmitted throughout the barn. But one of the most important part is getting the positions of all these cows. So as I said before, we're getting positions every second. And I started a movie here where the cow is moving. It's recently been milked. And you will see the small points will show where the cow has moved. It's not much happening in the beginning now. So I'll talk a little bit about the sensors. So each cow has a tag on its neck connected by a collar. So it's a small kind of tag. It's a sensor in itself, really. And then you have sensors up in the ceiling as well. So you can get the location of all the cows. So this is an ultra wideband system. And so we can follow the movement of the cows. And here we can see that the cow is starting to move quite a lot. It's going into the feeding area and trying to find something to eat. So now it has stopped to eat a bit and standing there. So this system has been developed for the farmer to check how much the cows eat and how much they sleep, if the cow is ill in some way, if it's in heat, and if it's ready to be inseminated. But what is new here is that we're using it to look at social interactions between the cows. So I'll forward the movie a bit, but you can see that we can see how the cow is moving around, what it's doing in different parts of the day. And we can do that for all different cows in the barn, but that would just be too messy if I did that here. So it's an already existing system, and we're just downloading the data from the two barns, really. Okay, so this is a photo of all the senior staff of the project. And it's taken earlier this year in Uppsala. And the reason I show this photo is for you to get an idea of the different competencies you need for a project like this. So going from left to right, we have Murud Alam. He's a statistician and an associate professor in microdata analysis at Dalarna University. And Anna Skarin is a senior researcher at SLU. She's been working on analyzing GPS data and movement data on reindeer for about 20 years, and her expertise in how to analyze animal movement data. Fred Fixe, he's a senior researcher in quantitative genetics employed at Växa Sverige. And then we have Per Petsnisen. He's a researcher at RISE, the Swedish Research Institute, and he has an expertise in precision livestock farming and animal behavior. He plays a major role in the project. He has a responsibility for collecting all the movement data, and he has previously worked a bit on social interactions of cows as well. Okay, so this is me with a beard, and I'm the PI of the project. This is Tariq Halassa. So he's a professor in animal welfare and disease control at University of Copenhagen. He has actually been involved this year quite a lot in COVID-19 modeling in Denmark, where he has been part of an expert group of modelers over the last few months as a support to the Danish government. So, I mean, there's lots of connections and similarities between this project and the modeling of COVID-19, social interactions and disease transmission, which is quite interesting. And it's quite easy to explain why this project is important. Volker Krumker, he is a professor at University of Copenhagen as well. So he's the expert on transmission of bacteria and mastitis among cows. He's one He's the one responsible for collecting all the bacteriological data and also supervisor of one of the PhD students. And then last but definitely not least, we have Nathalie von der Leer. So she's the communication officer on the project. So it's really nice to have your own communication officer on a project like this, and it's definitely needed. So that's the senior staff. And we've been working on the project for a year now, and most of the time has been on focusing on collecting the data and recruiting junior staff. So Svenja Wodstra, she is responsible for collecting the bacteriological data. She's a vet and is focusing on the infection dynamics of mastitis. Maya Guzman, she has developed a simulation program for disease transmission of mastitis and will incorporate the knowledge that we gain on social interactions and disease transmission into a program so that we can predict different measures you can take to protect the cows from different from the disease. And we have also recruited 4 junior researchers at the CLU with a background in genetics, in applied physics, in behavioral sciences, and in computer science. So before I continue, any questions so far? If you have any questions, this is the time to ask. If not, then I'll continue. Fine. So this first year, we have mainly focused on social interactions and disease transmission. And the planned papers we have for the next 6 months, the first of this one is the one I will present today, is where we find missing data. So where there are no signals, we lose the signals of the cows for some reason, and understanding the data quality is essential to be able to understand the rest of the analysis that we do. But the second paper we plan is what kind of characteristics of a cow determines the number of connection it has with other cows. The third one is where does a cow like to be in the barn? And we also need to do a sensitivity analysis before we do the network analysis. So we're going to look at how sensitive the social network analysis will be to missing data. And we will also look at if we can cluster positive from negative behavior between the cows somehow, just using the movement data. And for this, we will use observational studies as well. So one of the postdocs will actually be in the barn and watch the cows and then see different kind of behavior and how that connects to the movement data. And the sixth paper, perhaps the most important one, is the transmission dynamics of the pathogens. But the one I will present is the first one. So where do you find missing data inside the farm? So we're looking at 2 farms, and I will present the results from the Dutch farm. Then we have chosen 59 cows, and we have chosen 6 days with 2 weeks between each day. So it's almost over 3 months. And then we want to look at where do we have the variation within the barn. So where do we lose a signal within the barn? But we also want to look at if there's any variation between the cows. If one cow happens to have bad sensor for some reason. And we need to separate these patterns somehow. So this is a trajectory from one of the cows over one day. The blue spots are where we're getting signals, and the red ones are where we're losing the signal. And we're losing it for more than 5 seconds. So that's the kind of signals we want to see where we're losing it. So now you could imagine if one cow has a bad sensor and it likes to be in a certain place of the barn, one might detect this area in the barn as having being bad and giving bad signals for some reason. But that's just if you look at the raw data. But we want to separate the effects of the cow and the spatial effects. So how do you do that? Well, you can fit a spatial model, and we fit a spatial linear mix model where we fit both spatial effects and individual effects. And then we get this kind of map. So green means the signal is really good, and red means the signal is bad. And the white areas is where we have missing values. So one can see that, okay, this is we've been able to smooth the data. So we're getting rid of noise, and we can see that some clear patterns. First of all, there seems to be missing data along the right-hand wall, and especially in the top right corner. And this barn has a milking robot as well. And when the cow goes into the milking robot to be milked, we lose signals as well, which is perhaps not very surprising. But that's something we can see from the analysis. So here we're losing a signal, and up there, especially in the corner and along the wall, we're losing signals. And we're getting effects of the different cows as well. So we can see that, okay, here there seems to be especially 2 cows where it seems to have worse colors than the rest. There's one losing 4% of the signals and one losing 10% of the signals. So the conclusion that we draw, okay, we can show where we're losing the signals and which cows that have bad tags as well. So how do you fit a model like this? Well, we use a package called HLM in R, and something that we developed in 2010. In 2015, we included this kind of spatial modeling. And the model that we actually applied to analyze this data was a conditional autoregressive model. And that's something you can do in HLM package. And I will show you just some brief R code. So using the library HLM, you specify X1, which is the design matrix for the individual cows. And then we have Y1, which is the observed proportions of missing values within grid squares. So what we did with the map was that we separated into 20 by 20 grid squares and looked at what was the proportion of missing values within each grid square. So Y1 is a proportion of missing values transformed to be normal. Z1 is just an incidence matrix, not giving that much information, but the most important part is this grid map. So that's specifying which grid squares are neighbors to each other, and so you can borrow information between neighboring grid squares. And then we can do the spatial smoothing, and we can fit the cow effects as well on top of that. if you want to know more about hierarchical generalized linear models, which this HDLM package is based on, I produced a book in 2017 together with Yongju Lee. So he's a professor in South Korea who worked with John Nelder for many years. So John Nelder is one of the greatest statisticians of all times, who developed the idea of generalized linear models, who produced a paper together with Yongju Lee and Judith Pavitan at Karolinska Institute in 2006. And our 2017 book is an applied version of that, really. So there you can see some ideas of how you can do this kind of flexible modeling. So this is basically, just to sum up, what I wanted to show today, giving some insights to the research that I've been doing. This is my contact information, and we have a web page that you can follow as well, which I will try to show now, if I'm able to do that. Let's see. Yes. So this is a web page that we have. And what we want to do is to improve both health and production in dairy cattle. So we want the cows to have a better environment among the other cows. And by doing that, we want to increase the production. So ending up the talk, I want to thank my collaborators, of course, and also Formas and the Bayer Foundation, and for you to inviting me to this workshop. Thank you so much. And if there's any questions, I'm happy to take them, please.
Okay, so are there any questions from the audience? No?
If not, I can start. I actually have one technical question and one, well, not so technical maybe. I'm curious, well, I realize a barn maybe is a well-monitored environment, so you have a lot of information, but assuming it was less well-monitored, let's say, do you have any, or what sort of approaches do you have to imputing missing data? In your example, it was only like a single central spot, so not that much, but.
Yes. We actually do have some cows that have quite a lot of missing data. So what do we do then? And there are different kind of methods that we're looking into. And well, what we think we will use right now is just a cubic splines kind of fitting, using the information that we have, just smoothing the data that we have and filling in. And we hope that that will be enough to be able to do the social network analysis. But that we will do this sensitivity analysis as well and check that is sufficient. Yes.
Very interesting. And you touched upon maybe, my next question was, can you go on ahead with these sort of models and predict different scenarios? So if let's say if you were interested in improving the cow's environment or introducing some perturbation of some sort, who knows, within reason, could you then run scenarios without actually moving the cows, so in a way
yes.
to.
So this postdoc Maya at the University of Copenhagen, she's actually doing exactly that kind of simulation program.
Cool.
Looking at, okay, where do you put the water troughs, where the cows go and drink? That will affect the connections the different cows have, and also the disease transmission. And that's exactly what she wants to simulate. Yeah.
Interesting. And I guess this also has implications for our current predicament, but that's on a side note.
Yes, it will be very interesting to see how well we're able to model the disease transmission. Yeah.
Thank you.
Any more questions? I can maybe ask a short one. Do you will you only have data from cows inside, or will you also have like cows that live outside?
We're just looking at the cows inside.
Okay.
So, but during the summer period, the cows have the possibility to go outside for 3 months of the year. So according to the legislation in Sweden, the cows must have the possibility to go outside for at least 3 months of the year. Which means that we lose the signal when they're outside as well, which is a bit a complication to the problem. But we do not, so far in the project, we will not follow them outside now.
Thank you. All right. If there are no more questions, I guess we can thank Lars and also all the speakers of today. Maybe we can have a small applause. And yeah, that was the first day. I hope we will see you tomorrow again at 8:30, I think. No, I have to check again.
8:30 tomorrow.
8:30, yes. Or even better, at 18:30 for our social event, which will be in Gathertown.
All right. Welcome back, everybody. It's a beautiful sunny morning here in Gothenburg, at least. I hope it's kind of decent weather wherever you are as well. Welcome back to the workshop. I just wanted to quickly point your attention to two things. So we have a hashtag for this workshop. So far it's mostly been me and one other participant tweeting there, but feel free to use that if in case you want to tweet anything about the workshop. Yes, and then one more thing about Zulip. So yesterday, I think it worked to a certain extent that people posted questions in Zulip, but feel free to ask questions there or ask them in the Zoom chat. We will also monitor that. Or if nobody has any written questions, you're also free to just raise your hand and we will try to see that and ask you for your question. And if you have questions after the talk, then Zulip is maybe the best point or the best place to go for more further questions. Okay. Then, yeah, then I think we should start with our first speaker for today. That is Gunnar Cedersund, who is the head of the Integrative Systems Biology Group at Linköping University. And Gunnar is actually a speaker at the MBM Workshop for the second year in a row now. We're very happy to have you back. And after last year's workshop, we had a little talk at the end and realized that Gunnar has a very, very wide range of interests going from sports to the arts to science. And one of his passions is combining all of this into one product in some sense. And today, I think we will learn a bit more about how he combines all of his interests together with his interest in modeling the human body and its organs and the so-called digital twin technology. So we're very excited to have you, Gunnar. Just like yesterday, maybe 2 minutes before your time runs up, you will hear something like this. So you know that you have 2 more minutes, roughly.
Yeah.
All right. Then I will stop sharing here and you can start your own sharing.
Good. Let's do it there. So, and share computer sound, very good. So, let me minimize this and let me change to the laser pointer. Okay, so hello everybody. I'm very happy to be here. And as you said, for the second year in a row. And It's true that we talked after the meeting last time and discovered that this might be a nice workshop, which I enjoy very much, both this year and last year, for presenting a little bit wider view. So now I will sort of follow in the footsteps of John Hasenauer yesterday and William Lövfors, who presented detailed things about how to fit models to data and how to quantify it. And now I will talk about validation, integration, and also usage of these type of models. And I will do this centered around the concept called M4 Health, Models for Health, or mechanistic multi-level, multi-timescale, and multi-species models. So we construct these models by laying the puzzle of the human body, all of the different types of data, into multi-organ, multi-level models, which should be useful for healthcare, drug development, ordinary citizens that either want to stay healthy or want to use it in their creative expressions. And it's all centered around the concept called digital twins, which is a computer copy of something. And I will move from methods to applications. And here is the logo of our spin-off company, which we launched last year, where we actually turned this into a product as well, so people can use it. And here you see a digital twin of a cedar tree. And why is it a cedar tree? Well, it's because the name of the company is Sund, which means healthy or doing things in a sound way, which is the last part of my last name. And the first part is cedar, which is a cedar tree here. So this is my little name, and a digital twin here of a cedar tree. So I'm at Linköping University, which is also in Sweden. And this is the logotype of my research group, which has two components. The first is that it's a box that is not a black box. You can look inside, so it's a mechanistic model. And we also keep track of which predictions that are well determined. And nowadays we also do hybrid modeling, so we combine these models with black boxes. Maybe it should be updated at some point, but this is the logo now, and this is the one slide summary of my presentation. And I will basically describe the journey of the 20-year modeling journey of modeling all of the human organs. So here you see papers where I'm a co-author of all these papers. And we have now started to combine these more and more into an interconnected model that become patient-specific by adding patient-specific data from sensors and electronic healthcare records. We also started to do a software interface for this. And again, we do this by formulating our mechanistic hypothesis into ordinary differential equations, typically, which we either reject because they cannot be fitted sufficiently well to data, or they can be fitted to existing data, but then we want to do predictions to test if they can also predict new data that they didn't see or that didn't even exist. And once we've done that sufficiently many times, we have a system for incorporating that into the whole body picture and to simulate and use the models to see how we can go towards healthy from, for instance, type 2 diabetes. So here you see in DASH, you see there is no data for this. There is data for this because these are already well characterized, but this is a particular patient where we is to the blue one upon a certain type of drug. And we developed these models essentially in the way that William explained yesterday. So we have different hypotheses for how the system works. We have, in this particular case, in the paper that we published 2010, we had 4 hypotheses that could explain the original data set and overshoot. And then we did one of our 3 key tricks. Keep track of the well-defined predictions and look for the ones that are different. And then we know that no matter the outcome of the experiments, one of them will fall. And then in the same way, we kept going back and forth between the computer and the wet lab many times, but we did it for different reasons. So here it was to identify the parameters, and then we should measure something as uncertain as possible. And here it was to validate the model. There was only one remaining. Back and forth 5 times, so we give this the Systems Biology Index 5. So just an example of when we had a single model that was actually rejected, so we had to refine that as well. So here we have insulin binding to the insulin receptor, and we're looking here at this state here, the internalized dephosphorylated state as a function of time. And we see that it's a prediction with uncertainty. And the prediction is that 45% is internalized. And this was achieved in a sequence of steps. First, collecting all the parameters, and then characterizing really the boundaries of what is the lowest possible value that you can have, still be consistent with existing data. And this was 45% or more. And experimental testing showed that it was less than 5%. And then we already knew that this had to be a rejection of this hypothesis. So we had to reformulate it and do a validation again. And I won't go through the detailed methods of this, but here are some of the key publications. But I do want to make one comment. Don't use structural identifiability analysis or sensitivity-based methods for identifiability analysis. And I can argue afterwards why if you don't agree with me. So all of this led up to this minimal model for the insulin receptor activation and activity, which is a part of the overall insulin signaling and how it goes wrong in diabetes in fat cells. And this is exactly what William explained yesterday, based on these type of data, where you have red and blue corresponding to healthy and diabetics. So intracellular response. And then we have, as I said, a system for connecting this with the whole body level and the organ-organ crosstalk, so we can describe meal responses. Again, you see comparison between experimental data and simulations with uncertainty. This particular model was actually approved by the FDA. And another high quality model that we made use of was a weight change model. And this describes then changes over a long time scale, like months and years. And this is all the way down to minutes and seconds. So it's multi-time scale, multi-level, because it's whole body to intracellular level, and mechanistic. So how do we do this connection? Well, the key thing is to identify modules. And then you want to have models where the inputs and outputs are known. And then you can require that for these known inputs, the output of the module should be within an acceptable range. So if you have glucose and insulin in the arteries flowing into an organ, for instance, then glucose uptake has to behave in a certain way. And you can measure these by doing arteriovenous difference data, which are data where you collect the arteries and the veins at the same time. But it can also be simulations from the top level model. And if you have these constraints, then each submodel, developing models for each submodule becomes an isolated modeling problem. So you can have many different model versions, and all of these here will be compatible with top level model version A, as long as they are compatible with this input output. So you can replace them in a relatively seamless way with each other, depending on the question. And apart from these metabolic type of models, we also looked at liver function, neurovascular coupling in the brain. We have a sequence of papers on that, and blood flow in the aorta, muscle, pancreas, spleen, and so on. And all of these are now more and more interconnected. So what about personalization? Well, our patient 0 looks like this on the outside, and on the inside, above you see his fat deposits, and below you see his muscle deposits collected via an MRI. And we put various sensors on him, like a blood sugar sensor. And here you see comparison between data, which are the lines and simulations, for estimation data and validation data. So the model, the patient-specific model, is trained on the breakfast response here, which you see here. And it's validated then by doing a prediction, just looking at his diary, what did he eat for lunch, and then see, can we then correctly predict how the blood sugar will change over lunch. If we can do that a number of times, we go on to use the model for predictions with uncertainty, where we keep track of if we are close to the usage of the model where it was validated, then we fulfill the FDA criteria for it, that you can use this model in certification. In the same way, we collected data for heart and blood flow using 4D flow MRI, stress responses, and so on. What we are now moving towards is to use this model in the clinic, first for better pedagogics, so to convince the patients, explain for the patients why they should have a high compliance or follow the treatments or do preventive measures. And after we used those type of things, which are the low-hanging fruits, we will also go to personalized diagnosis and treatments, which require more certification. So this technology was launched as a beta product last year at Almedalen, big Swedish event. And it's led to keynote presentation at, for instance, NIH. And if you want to see some of the functionality of this, here is a link to a YouTube video. So a little bit more about this logo. So you see a digital twin illustrating that we are helping people make decisions, and it's and that we want to tell stories. So we try to do sound science, Sundforskning in Swedish, and help people to tell themselves sound stories, and thereby inspire them to make sound medical decisions and overall to lead a sound life. So if you take a particular such story, here we have, let's use the case of NAFLD, which is liver fat. It's connected to obesity, diet, dyslipidemia, and it's a risk factor for liver complications and stroke. And if you get a stroke, or as you see in the images here, you get COVID-19, then you typically end up in the ER, but then before that you can do prevention, and after you can do rehabilitation. And then by simulating different scenarios, we can show how the liver fat is getting worse or better depending on the person's diet, exercise, etc. And health conversations that we have implemented here in Linköping has shown that it's already lowers the risk to have these conversations about these type of things without any other things than just a conversation explaining these things, lowering the risk of a cardiac event with 30%. And we want to magnify this effect by then showing patients that if you go here on an LCHF diet, so low carbohydrate, high fat, you will reduce your liver fat by almost 40% within a two-week period. So here we see again comparison between data and simulations for our model on this. And then we want this to follow in the patient journey, so that you can have wearable sensors that you bring home from the conversation, so home monitoring, and then you bring it back to the cardiologist or hepatologist or some other specialist. So this then, we want to help in this patient journey by having this type of simulations where here you simulate various interventions. So here you define the schedule. I want to go on this diet. I want to start exercise, so take this drug. Here you see yourself, if you have an MRI image, otherwise one will be picked. And you can rotate it, you can zoom in on different parts, and you can also look at different organs. You can click on an organ and see what you have inside. And here is then a liver image, and all of the white things there, were quite many, they are fat. And then you see that by doing this diet, you would reduce it quite a lot. And then by clicking again on all of these things, you see how these white dots disappear. So your liver is essentially healing a relatively short period of time. And apart from that, you can also show risk factors. So here we have different types of risk factors. Some you have measured, and some you don't know. And the ones that you don't know, we developed a system for imputation. So now you see that all the values are filled in. And this is done in a certain way that I'll explain on the next slide. And then you can also calculate the risk. So the technology for doing this type of combined hybrid models is to have a two-step approach where we first have blended hybrid modeling, where the mechanistic models become population models using nonlinear mixed effect models. And these generate biomarkers, which are new model inferred, which together with omics type of biomarkers go into the ultimate machine learning model. And another approach for this is to do simulation of such hybrid models. And then again, we use this large UK Biobank and other really large clinical studies, imputation to get values that you don't have, either after the simulation or after the measurement. And then risk calculation, which also comes as statistical models from the large clinical databases. And then the simulation shows the trajectories of whatever you can do mechanistically. So in that way, you can simulate more variables, and you can also do simulations with fewer measurements. This should hopefully be published relatively soon. Looking at the timing of this. Last year was the launch event of this, and now we are creating more healthy digital twins by doing detailed clinical studies where we're measuring as much as we possibly can using as many devices as possible on each individual so that you get patient-specific models that are unique for each patient. We're presenting this, publishing all the submodels and the interconnections. We'll release a new version of the software, and next autumn we will start to implement this in this health conversation. And if that works fine, the digital twins will follow the patient's home for home monitoring, and then in various types of patient groups also when they come back to the clinic, type 2 diabetes, stroke, high blood pressure, and so on. So if you now look at what this can be used for, it's to do the challenge that Jan Hasenauer mentioned yesterday, that we need to integrate all sorts of different types of data, mechanistic knowledge, mechanistic studies that come out, the big clinical studies, personal data from activity clocks and online forums and diaries and electronic healthcare records, and to create these digital twins, which are created for each person, updated when new knowledge comes in, can visualize how things are now, and can simulate future scenarios. So that you get to, as a doctor, you get a quick overview of a patient's status. You can dig deeper if you want. You can have use it to tailor treatments for patients and improve communication. And from the patient perspective, you get more preventive and personalized care, patient and knowledge-driven care, patient-specific tailored care, and in general, more motivated and healthier patients. So another positive change is for drug development and for 3R, replacement of animal experiments, where we work together with AstraZeneca to basically help transform their drug development system, which, as for any drug development company now, is a linear pathway where you screen many, many drugs on cells and then on various animal systems before you move on to humans. And we want to do this knowledge-driven using our interconnected M4 models. So each experiment that you do, either on cells, animals, organs on a chip, or humans, brings new pieces to the puzzle, the understanding of the disease, which means that even failed drugs, unlike now when it's just wasted money, now they will give valuable data back to the preclinical phases and the design of new drugs. And this also means that we have a pathway here where we can go directly from cells to organ on a chip to humans. And here we see an example of this. So here we see an organ on a chip where we see this type of meal responses. So we have a little mini human here, a human pancreas and a human liver. And you see meal responses. So there's glucose and insulin interplay here. And you see comparison between data and simulations as usual, also for validation data. But here you have a 48-hour meal response. And then that is because the volumes are different on the chip. For experimental reasons, you can't have exactly the same volume proportions. But what we can do is that we can train the model, we can validate the model for the chip, and then we can rescale the different organs and different proportions so that they get human proportions. And then we see that we get back to 4-hour meal response that you get in humans. So you sort of can do the experiments on the organ on chip, and then you translate it using our models. You rescale the organ on chip to the full human. Again, prediction with uncertainty. And in the same way, if there are people that really love animal experiments, don't worry, we have something for you as well. Here we have a corresponding model for mice, multi-level, multi-timescale, so we can simulate long-term weight responses for estimation data, validation data for meal responses in response to different interventions, and intracellular things on different timescales, all of this. But the problem with animals as they are is that they aren't perfect models for humans, also not. So type 2 diabetes, it develops by having an increased insulin secretion over time, which leads eventually to beta cell failure. But if you look at available animal models, you have developed one which has the insulin, increased insulin secretion and the insulin resistance in place, but their beta cells never fail because they live too shortly, probably. And then you have some mice where you destroy the beta cells, but they don't have the development of the insulin resistance and the increased insulin secretion, and the failure is not because of that. So what we have done is to is to develop in silico mice, which can do all of the things that you have here. They are validated for the blue mice. So they have the insulin resistance, but then we have given them human beta cells in the computer so that they will fail after a while. So you sort of have, you can improve animal models as well in silico. And here is one of my new favorite projects, which is that I'm also a concert pianist, and I played all of the ballades by Chopin and 16 of Beethoven's 32 piano sonatas. And I want to illustrate, I want to use this to illustrate what happens. So here we see some piece of music where we take an abstract representation of that. So we identify what happens in the music. And then I collaborate with, for instance, Julia Bengtsson, who is a very talented ballet dancer and choreographer. So she earlier this year she had a sold-out performance in Carnegie Hall with her own choreography and her own dancing, which is super cool because it's maybe the biggest, most famous concert hall in the world. So we do a choreography together where I'm playing and where she's dancing, but where we decide what she dances together. So here you see us practicing, and this is exactly this music. That was the beginning of the blue, and now comes the green one. And in the same way for the Beethoven pieces, here you see the beginning of the fugue in Opus 110. And now we practice over Zoom, so you see this is at our home. And then we use pose detection and GANs, which is a type of AI, to get corresponding dancing in the digital twin, so you can see them move in the same way. And a trailer will be released of this project within the next few months. So then you will see how you can listen to more of this. Since the pandemic, other things have changed. We also started some projects where we are starting to look into how we can introduce COVID-19 into our twins. And even more, we have started with epidemiological models where we use the governmental models and data, and we have meetings with them to help them to have a sound combination of different disparate data, do hypothesis testing and predictions with uncertainty, and in the end to have multi-layered models where you have agent-based models that can be fitted to data, and where each agent is a digital twin. And this can help with societal actions like monitoring of spread, predictions of social legal interventions, and to identify high-risk patients, tailored treatments, and so on as before. So to sum up, the long-term vision here is to have a personalized, patient-centered, interconnected healthcare system, where already at birth you maybe donate some cells, you donate some data, you create the first version of the digital twin. And then you add new cells when you need to, and you can then do experiments on your own cells in a little mini-you, which you can translate to your digital twin. You can add new data from smart sensors, from meetings with clinicians, throughout your patient journey. So you can interconnect the now very separated, disjointed healthcare system. You can also use it to tailor drugs for yourself, and again, for music and arts and gaming and all sorts of things. Just to wrap up, I want to thank my group, which are doing all of these different things together with lots of different collaborators. We have been very fortunate to get a lot of funding in the last couple of years. And just to give an example of how dedicated they are, yesterday there were 7 or 8 people that stayed up until maybe 8 in the evening to work with some of the new slides that we had for this presentation for today. So I'm very grateful for my research group. And that was it for my presentation. Thank you for listening.
Thank you very much, Gunnar. So, do we have any questions? There's nothing on Zulip right now. You can also ask through Zoom if you would prefer that. Otherwise, I have one. Oh yeah.
Yeah, I didn't find the raise my hand on time. So sorry. No, at a certain point, you said don't use structural identifiability.
Yes.
And I'm very unknowledgeable in this field, but and I see actually Johannes now appearing on my screen, but he can maybe confirm. But don't we use this quite a lot in our group?
Yeah, many people do it. And from a theoretical point of view, it's very beautiful. You use this differential geometry and abstract algebra type of things. It's very beautiful mathematics. But to find out about the identifiability of the system, it's completely useless. If you have, so I have some examples I can show you where if you have a really big model with 60, 70 parameters, and you have one measurement variable, then you can have the identifiability analysis can show that you have 4 parameters that you can't identify, but the other ones you can. But in practice, with one measurement variable, you can probably identify 4 parameters instead of 58 parameters. And it's neither sufficient nor necessary. So it's not sufficient because of the example I told you. It doesn't guarantee anything, but it's not even necessary because you can have structurally non-identifiable parameters that are still practically identifiable. And this happens if you have constraints in parameters. So I published this in my 2009 paper, but somehow it hasn't gained traction. But if you have just 2 parameters that are summed together, then they will be, and they always appear as a sum, then they will be structurally non-identifiable. But if you know that they are bigger than 0, then you can identify the sum of them, and therefore you can have an upper boundary. And if you also in that sum have an additional parameter, you can also have a lower boundary. So it can be as identifiable as you want, even though it's structurally non-identifiable. So it doesn't tell you anything.
Okay, thank you.
Hello, could I ask you a question?
Of course.
Yes, so hello Gunnar, do you hear me?
Yes.
Yeah, do you see me as well? I don't see you, but nice, very nice talk. And I was wondering how far, do you have any collaboration with Vårdcentral, as it's called in Swedish,
yes.
Smaller hospitals, and it's in use, so to speak?
Well, we will do the first clinical implementations next year, next autumn, and then it will be in the Vårdcentral, in the primary healthcare centers, where they do this health conversation. And we are preparing for this now. So we're having the design of the interface and so on is done in dialogue with them.
That's pretty amazing. It's so yeah, exactly. No, it's a lot to take in. It's so useful all of a sudden, systems biology, when you do it like this. It's nice.
Yeah, and I think it has been too much of a focus on just explaining everything in the cells and not bringing it into the clinic in the systems biology community, I think. So that's where I would try to position myself, that have systems biology in cellular stuff, but still make it useful in the clinic and for end users.
Could I ask another question, the organizers?
I think we should move on to the next talk, but you feel free to go to Zulip and ask questions and discuss there, or later during the speaker room when Gunnar will be back for more discussion.
Okay, yeah, thanks.
Very good.
Okay, thank you. All right, so we come to our next speaker and to the last speaker of this first block today. That is Carl-Joar Karlsson. He's a PhD student at the Department of Mathematical Sciences here in Gothenburg. And among other things, he works with game theoretical models and studies the spread of disease, which is very timely now in times of COVID-19. So yeah, Carl-Joar, are you ready?
I am ready, thank you. So can you all hear me?
Yes.
Loud enough, thank you. Okay, so let me start the screen sharing. I think this works, right? Okay, cool. So first of all, thanks to the organizers for organizing this wonderful conference and giving me the opportunity to talk about my projects in game theory and disease spreading, which I've had the honor to work with Julie Rowlett on, my supervisor. So indeed, when we started on this project, we were looking at the current situation with the COVID pandemic, and we thought about the behavior in the population of humans, because that can be sort of described by game theory, which we knew about. And then on the other hand, there's a lot of mathematics in the dynamics of the disease. So our question was, can we combine these two things? And what the outcome of that in initial brainstorming is this paper, which is open access, so you can download it and read it following this link. I want to show some details on this, and I will start with the disease part and then go to the game theory part and see how they combine later in the talk. So first, one of the most well-known models in disease spreading is the SIR model. It's a so-called compartmental model, which says that on the population, we can assign labels to each of the individuals. Either they are susceptible to the disease, or they are carrying the disease, they are infectious, or they are recovered from the disease. And the dynamics are shown here in the equations. And there are two parameters that decide the dynamics. One is the transmission rate, how quickly is the disease carried from infectious to susceptible. And then there's the recovery rate, gamma. And now there's a simpler model, which we will also work with, which is the SIS model. And that simply has no recovery in the dynamics. So if we're susceptible and then catch the disease, then we go back to being susceptible again. We can say that there's no immunity to the disease in that model. And because there are only two compartments in the SIS model, we can very easily simplify the equations, and we get one single equation describing the entire dynamics. And you see here still two parameters, beta and gamma. So that is the disease spreading part, the dynamics of the disease in the population. And now let's look at what game theory has to do with all this. So first, the basics. We consider game theory as something that describes the relation between choices and outcomes. So in this case, we have a so-called normal form 2x2 game, saying that there are two players having two options each. So imagine now the player in player one, which we call me or I, is in the column here. So when I choose to cooperate, then we see what happens there. And there's a player on the top row that can also make a choice between the two options. So if I choose to cooperate, what this table says is that if the other cooperates too, I get a payoff, which is just a number R. And if the other defects, I have a payoff S. If I choose to defect, on the other hand, I can have a payoff T or P, depending on what the other player is doing. And now we need a description of the population. So we can imagine now that we have a big population where each pair of individuals play this game. And we, as individuals, do this repeatedly. So we pair up with randomly with a random chosen other individual, and we play this game, and between each game we can change our strategy, but we do so with a probability. So let's say that we have a probability X that we play cooperates. So we can reduce the previous table to this one because the game is symmetric, so we can just swap the labels on the two players. And we compute averages because in the long term, that is what decides sort of what the payoff will be if we just continue with our strategy. So if I cooperate, I get this as my average payoff, and if I choose to defect, I get this. But we want also to see what behavior sort of due to this payoff structure. So imagine now that we can also change strategy. And to do so rationally, we would like to know what the payoff becomes if I change my strategy. So to incorporate this mechanism, we use the so-called replicators equation. And that is to say, if my strategy has a payoff, f subscript x, which is greater than the average payoff, which is in the angled brackets here, then I increase the probability to play that strategy. And that is what this equation describes. And with this normal form 2x2 game, the right-hand side looks like this. So this is what we want to combine now with the disease spreading model. We have a individual mechanism that affects the whole population. But how do we actually do this? That's the question. And in that we were inspired by this paper by Poletti and collaborators. So if I'll just briefly go through what they, how they constructed this. So recall the SIS model. We had one single equation for that, and the parameter beta is the transmission rate, and we can let that depend on the actions in the population. So we imagine that there's a portion now of the population that has a transmission rate beta C, and that the remaining has a transmission rate beta D. And in the equation, we simply put in the weighted average of these two. And it's time dependent because the portions are not static. They evolve according to some sort of replicators equation. In the work by Poletti, they assume that the derivative with respect to time of that portion is proportional to the number of infectious individuals, which is sort of to say, if there are many people out there which carry the disease, we're scared of that disease and we play cooperate for the good of the population. So that's their setting, and they find the asymptotics as t tends to infinity, which is pretty cool. But we found that we can do sort of a little better with some assumptions and using this normal form 2x2 game instead. And we sort of start with the same construction for the SIS model. That means plugging in the weighted average in the transmission rate. But we add to the derivative of x the payoff for the normal form 2x2 game. And we actually we're able to find explicit equilibria at large times. So this is what we have, and I will show the full set of equations and how we sort of simplify them a bit. So this is what we're working with the SIS model. I'll get back to the SIR model later. And the second equation is what is really interesting because it tells how the cooperation develops over time. And there are two parts of it. One is the so-called, so to say, awareness of the disease, which is to say if beta D is bigger than beta C, I reduce the transmission rate by cooperating. But in doing so, I also need to consider my payoff in the normal form game. And what we do is we assume that there's a so-called prisoner's dilemma, which is a notion in game theory, on the immediate individual payoffs. And therefore, there's a competition between two mechanisms. One is the awareness of the disease, which affects the whole population. And on the other hand, there's the, so we can think of it as sort of a self-interest, the payoff that's immediate on our choices. And one tells us to cooperate and one tells us to defect. So now there's a competition between these two drives. And we want to study how this balance affects the equilibria. And with a couple of assumptions, we are able to bring these equations down to a simpler form. And most importantly, we introduce what we call a timescale alpha. And that tells us sort of how often we make choices based on either the normal form 2x2 game choices or based on the information about the disease. And what we then have at the end of all this simplification is this set of equations. And doing stability analysis, we identify the equilibria. And you can see here that there are 5 of those, but the 2 first are unstable. And this is for the SIS, we call it the SIS prisoner's dilemma system. And they all have different regimes. So if we consider what happens when alpha varies, we find for small alpha, there is no cooperation. The x is equal to 0. And then for very large alpha, everyone is cooperating. X is equal to 1. And there's an intermediate region here too. And you see that the equilibrium for the number of infectious individuals is static if we're not in this intermediate region. And in a graph, this is what it looks like. So following the red line, we see the number of cooperators, or the portion of cooperators. And if alpha is very large, everyone cooperates, and if alpha is very small, no one cooperates. And at the same time, as alpha grows, which is to say we more often make decisions based on the information on the disease, there is less infectious in the population. And we can also look at what happens now. So we have full control of this system and the equilibria, and we can play with the parameters. And most importantly, we want to find out what happens if we become better at reducing disease transmission. That is, if beta C is lowered. And we see here that then the number of infectious individuals also is lowered. So it actually tends to 0 if we become very good at not transmitting the disease. And so this is all for the SIS model. There's a totally different behavior for the SIR model, because the disease has its lapse and then it's over. So in the SIR model, there's actually only one stable equilibrium, which is at x equals 0 and I equals 0. So there are no infectious individuals. All are either still susceptible or recovered. And no one cooperates at large times. So this is pretty, this really highlights the mechanisms of cooperations in a very simplified manner, but the simplicity of the model is also the strength, because it's definitely shows like these regimes very clearly, and there's a clear interpretation, I think. We simply see that if the if there's immunity in the disease, no one cooperates. With the SIS model, no immunity, we find cooperation if this time scale difference is big enough. And we also see that the number of infectious individuals can be brought down to 0 if the disease transmission rate is very low for the choice to cooperate. So with that, I'd like to thank you all for listening, and I hope you enjoyed the talk. And I've put here a quote from the article so you can gain some inspiration, and I hope to receive some questions on it. So thank you all.
Thank you very much, Carl-Joar. Do we have any questions from the audience right now? I was wondering that now in times of COVID-19, and especially in Sweden, where people are not forced to behave in a certain way, so they can either cooperate with the guidelines or ignore the guidelines. Did you see any similarity between how people behave and what your model would predict?
I think first and foremost, we can compare different countries. So some of the countries that have had a lot of information to the public in general, to the citizens. In those countries, we've seen that people actually start wearing face masks and keep the distance and so on. So and they are also often very successful at reducing the cases of COVID. But my expertise is really on the mathematics, so I shouldn't state this as something obvious. There are more mechanisms behind what actually spreads the disease COVID-19, but we can see that there's some indications that this agrees reasonably with what different countries have as a strategy to mitigate the spread of the disease.
Okay, thank you. Any other questions?
I guess I have it's a very nice presentation. I'm wondering, kind of related to Felix's question, could you use this model or similar models to derive, let's say, strategies to influence the population, knowing the given behavior, and then you would like to maybe achieve a different behavior? I know it's a vague question, but
yeah, well, I can first of all state that this model is, of course, of interest in other fields than just disease spreading, because actually I've read some articles about like information, say that in social media, for instance, information can spread among the population based on, you know, like how many interactions there are between different kinds of individuals. And if we just find a system which behaves somewhat similarly, and we can give a reasonable interpretation to the payoffs, then the model would apply and we could analyze that setting. I hope that answers the question.
Sure. It's very interesting, the wider applicability. Thank you.
Great. Thank you very much, Carl-Joar. So if anybody has more questions, there's of course Zulip, and maybe depending on just a little thing, like the speaker rooms, originally we thought we would do them on Zoom, but due to the experience with the poster session yesterday, we will have it on a platform called GatherTown. So everybody will be in the same room kind of, and you can go and join local conversations. And that way also all the other speakers, not just the invited speakers, will have the opportunity to be present there and receive more questions if they want so, or if there are more questions, you can also ask them there on GatherTown later. Yeah, we'll take a short leg stretching break. Thank you very much to all the speakers of this first block. And we will be starting back up here at 10, so only 4 minutes, but so we don't push our schedule too much. See you soon. Okay, so let's start with the second block of speakers for today. And we will start off with Sebastian Persson. He's a PhD student at the Department of Mathematical Sciences, also here in Gothenburg. And he studies pathways and cellular processes through both dynamical modeling and also spatial modeling, and also has an interest in multi-level modeling and works at the Zrimec lab here in Gothenburg. So please, Sebastian.
Thank you for the introduction, Felix. Then I will just share my screen and hopefully you all can see my presentation. Nice. Then I will just go into presentation mode and I will start. So I would like to first thank the organizers for allowing me to present here today. And what I'm going to talk about is my latest paper, that is fine-tuning of energy levels regulates SAC2 via SNP1 dependent feedback. I will just see if I can find the laser pointer here. Or maybe not. Hopefully you can see my cursor. So this paper is published this year, so I recommend to read it. From my unbiased opinion, it's a very interesting read. But we can maybe start with the question that hopefully all of you ask yourself in the audience right now, and that is, what is SAC2 and why is it such an interesting gene to study? And the reason why SAC2 is a very interesting gene to study is because it's a part of the nutrition sensing network in yeast, that is normal baker's yeast. And the nutrition sensing network is basically what the name suggests. It's a signaling network that senses the amount of available nutrients like glucose and nitrogen. And then based on these levels, it correctly regulates the cell metabolism. And since the cell metabolism is highly central for cell viability, it's maybe not that surprising that a non-functional signaling network has been connected to diseases such as cancer, but also to such things as premature aging, which makes this very interesting to study and also highly relevant. And in this paper, we focus on a subpart of this network, and that is basically the glucose sensing network, which is not that well understood. And our hero of this story, that is SAC2, is basically a part of this glucose sensing network, as you can see in the figure here. So basically what we have is that glucose will affect the SNP1, that is SNF1, pronounced SNP1, I really don't know why. And then downstream, this will affect the SAC2 expression. So what you see in this image here is basically the previous knowledge about the SNP1, or rather the current understanding before our paper. And a consequence of this current understanding is that if glucose is decreased, then SAC2 should increase, because then SNP1 will not be able to drive this phosphorylation, and these dark blue arrows will dominate in the system. So if we would cut the glucose levels at time 0 in some data set, then this is kind of what we would expect from the SAC2 expression, that it should increase and then reach some new steady state, because we have these dark blue arrows driving the system. However, this is actually not what happens, because when we collect our experimental data of the SAC2 expression, we rather see that we have this expected increase, but then we have the expression level going to a decreased state and reaching a new steady state that is somewhat higher than the initial value. And I mean, this is not a unique occurrence. If we collect a data set of 124 cells, we can see from these quantiles, that is these dashed lines, that this trend is very general. We have an increase followed by a strong decrease. And given this current knowledge of the SNP1 pathway, this behavior is really perplexing. And in the paper, we kind of set out to try to figure out a mechanism behind this behavior. That is, we wanted to propose why it looks like it's doing. And one way to propose a mechanism is to go into the literature, read a lot, maybe do some experiments, and then propose that this is the mechanism. But since we have this time-lapse data available, we can do even better. We can do what Jan and Gunnar have talked about, and that is basically we can construct a dynamic ODE model and fit it to this data. So we basically want to formulate an underlying dynamic model, and then we want to estimate these rate constants by using some output y, which in this case is SAC2. And then we want to estimate our model to the data. And if our literature-based model strongly fits the data and also matches up with our experiments, we have a strong indication that maybe this mechanism we have proposed is feasible. At least we also know that it's capable of capturing the trend. But doing parameter estimation in our setting is really not that straightforward as for this single time-lapse series. Because as you might remember from the previous slide, we kind of had this time-lapse data. Because each gray line, which is 124 of them, corresponds to one cell. And there are two alternatives how we can do a parameter estimation. Either we can fit our model to this middle dashed line, that is the median, or we can have an underlying dynamic model or a state space model, depending on the problem, that will fit to each individual gray line, that is each yi. And I mean, from a mathematical perspective, both of these approaches are feasible, but the single cell modeling should be preferred, because the problem is that population average data, like the median, it can hide or mask dynamic features. For example, imagine if you have a cell expression that oscillates at a single cell level. Then if you aggregate all these oscillations from many different cells, you probably just get a flat line. And your model might perfectly fit this flat line, but it will not describe what happens at a single cell level. And that is kind of the problem. Fitting your data to a population average model might result in a model that actually does not describe what happens at the cell. Furthermore, fitting a model to the median data will not allow you to deduce sources of cell-to-cell variability, like what is this source of the variability, might it be due to aging. So therefore, we wanted to go for the single cell perspective. And we wanted to have an underlying ODE model that we somehow fitted to each individual gray line. But in order to do this, we have to account for the cell-to-cell variability. That is the difference between these gray lines in our ODE model. And there are different ways to do this, but luckily we can argue quite strongly from the biological pathway that the extrinsic noise is a dominating source of noise in the pathway. So basically, that difference in protein levels gives rise to the difference between these gray lines. And one way to include this difference in protein levels or this extrinsic noise in the model is basically to let the rate parameters k vary between cells. But we can also place a stronger assumption here. Because for example, if K1 for cell 1 is 5, then K1 for cell 2 should not be 5 billion. I mean, it's totally unreasonable. Because we are looking at a homogeneous cell population of yeast cells. So it's rather fair to assume that the rate constant should be somewhat similar. Rather, they should actually follow some multivariate probability distribution. So that means that the rate constant for cell 1 should probably be a realization like this one, and K2 should be another realization, and so on with K3. And if we can estimate this population distribution in a parameter estimation, we basically have all the data we need to fully characterize if our model is able to fit the system. So our goal with the parameter estimation in our case basically became to estimate this probability distribution. And luckily for us, we really don't have to invent the mathematics to do this, because estimating a distribution from the data is kind of, when you have different individuals, is a very highly common problem in the pharmaceutical industry, where they work a lot with the PDPK models. And here they use nonlinear mixed effects modeling. And just as a brief overview, the idea behind NLME is to first assume a parameters distribution of your parameters. And we assume the log normal distribution. And the reason why we assume the log normal distribution is that this kind of makes sense from the nature of the log normal distribution. Because if you have a random variable that is a product of independent identically distributed variables, you have a log normal distribution. If you have a sum, you get a normal distribution. And the rate constants, they are basically a product of different intermediate steps, but we also lump different concentrations that we assume to be basically constant. So all in all, we kind of lump different protein concentrations into them as a product, and hence the distribution should be log normal. And given this, we can finally formulate a marginalized intractable likelihood function, which we can use quite sophisticated optimization algorithms to actually optimize. So given this likelihood in this NLME framework, we could finally fit our models to the data to describe why the SAC2 expression looks like it does. But before we could actually fit the model to the data, we kind of needed to resolve this small problem of actually having a model. And just to bring it back to the problem in question, what we wanted to do is to take this current understanding of the SNP1 pathway and basically extend it so that we could explain why the SAC2 expression decreases upon long-term glucose starvation. And we had a hunch that this decrease in SAC2 expression was due to a feedback acting on the SNP1 pathway. And the SNP1 pathway is all these components, but also many more not shown here. And we built our first very simple model, and it fitted the data really well, confirming our hypothesis that this mechanism with a negative feedback on the SNP1 pathway is feasible. But we also kind of wanted to go further. We wanted to know what might be the source of this feedback and on which component of the SNP1 pathway might it act. So we went into the literature to try to answer this. And one thing that is quite known about the SNP1 system is that it kind of activates the cellular response to starvation. So when we have that external glucose levels are cut, then SNP1 will become activated. And this SNP1 complex will activate genes such as SAC2 and multiple other genes that will basically start recovering the intracellular energy levels because they will turn off energy consuming processes, express high affinity hexose transporters, the list is endless. So what we should have is that when we cut the glucose, basically the intracellular energy level should decrease because we're a drop, but then they should start to partially recover due to this SNP1 protein and others acting. And this recovery in intracellular energy levels should kind of correspond quite well with the decrease in the SAC2 expression. So we added in our model that we have a starvation response, so a feedback mediating component, that is this starvation, or rather intracellular recovery. And then we wanted to know on which part of this pathway might this one act. So we went to the literature and we also performed some experiments. And what our experimental data showed is that SNP1 is central for SAC2 expression in high and low glucose. That is, in order to actually have any SAC2 expression, we kind of need to have SNP1, which means that the feedback is likely to act via SNP1. Furthermore, to confirm this, it is quite strongly believed in the literature that SNP1 is regulated via energy levels. So that means that if we have this partial recovery in energy levels by the starvation response, then SNP1's activity or need to strongly express genes such as SAC2 should decrease. So we should have some inhibition error here, basically removing the need for having a strong SAC2 expression. So with this, we finally had our model structure. And now we had a modeling framework to fit it to the data. And the first thing we would want to see if this model structure is capable of capturing each gray line in the data set. Can we capture the individual cells? And this is the case, because here you see the individual predictions against the observed values. And you can see that our data falls beautifully around the identity line, which you can see on the trend line here, meaning that we have a good individual fit. But we can actually go further in looking if our model has a reasonable fit. Because the idea is that with the NLME framework, we estimate the population distribution of the parameters. And if we simulate cells from this distribution by simulating parameters, then our simulated cells should correspond well with our observed cells. And this is kind of exactly what happened, which we can see in this plot here. So we see that the simulated quantiles, which are the black ones, corresponds well with the blue ones. I mean, of course, there are some deviations, but overall, the general fit is quite good. So what we can conclude from this is that our model structure seems really feasible because it fits the data well. Furthermore, our model structure corresponds really well with the available literature and with our experiments. And all to all, this suggests basically that fine-tuning of energy levels, that is the starvation response, regulates SAC2 via SNP1 dependent feedback loop. And given this, I think it's time for some good take-home messages. And the first good conclusion we can draw from this paper is that SAC2 expression decreases upon long-term glucose starvation. And our systems biology approach basically suggests that this is due to a partial recovery in intracellular energy levels. And I should notice that I'm not the first one to show this in the literature because two of the keynote speakers has worked with time-lapse data and NLME, but at least this paper confirms that LME is a useful tool for time-lapse data when you have a dominating extrinsic variability and your system is not too large. And with that, I would like to give some acknowledgements. So I would like to thank SSF for basically funding my PhD. I would like to thank my co-authors Nick, Sveta, and Maria. This would have not really been possible without them. And lastly, I would like to thank the entire Swedish lab for basically providing an awesome research environment for doing your PhD. And with that, I'm happy to take any questions from the audience.
Thank you very much, Sebastian. Are there questions in the audience? I was wondering, so now that you have this relatively small pathway model, are there plans of embedding it into a larger surrounding of a bigger model to have it as a part of a, like, maybe in the direction that Gunnar also described, like embedding smaller module parts, models into a bigger context?
Yeah, that would be interesting, but then we would probably have to look at the average of the model, because the problem with building these single cell models larger at the moment is that first paying the parameter estimation is quite daunting because it takes quite an amount of time. And also, since we are kind of estimating a distribution with a full correlation matrix, we have kind of an explosion. With each parameter we add in the model, we potentially increase the number of parameters by a square law. So it would be ideal to extend it to further modules, but then I also think it would be ideal to extend it to a further larger scale single cell model. But I think that in order to achieve this, some research is going to be needed on the methodology, for example, by knowing which parameters you really don't need when you estimate the population or the distribution rather.
Okay, thank you.
I also have a question. If there's nobody else, I shouldn't take from. Very nice, Sebastian. I'm curious, you mentioned that the dominant extrinsic noise is difference in protein levels. I don't know much about single cell, but that's super interesting to me. Does that reflect the transcriptional pulsing that is known to happen at individual cell level, where over time you would see this fluctuation in transcription levels?
Oh, that is actually a really good question, because there are two sources of noise you can put it into. You either have the intrinsic variability, and that is due to the fact that chemical reactions are inherently stochastic. You actually need to collide in order to have a reaction. And this oscillatory behavior we see, I think it in the long term might be due to the extrinsic, but it's very hard to see because there are very few of these species where you can have this oscillatory system. So it might also be due to the fact that molecules have to collide. It's probably due to extrinsic because molecules have to collide and they take different paths, and then you might reach the behavior. But it's really tricky to say because this is these systems become quite complicated quite fast.
Yeah, very interesting. Thank you.
Nice. Okay, then thank you very much, Sebastian. Then we will come to our next speaker, and that is Ida Larsson, who is a PhD student at the Department of Immunology, Genetics and Pathology, very long name, at the Uppsala University. And she works with computational systems biology of neural cancer at the Nylander lab in Uppsala. Welcome, Ida.
Thank you. Can you hear me?
Yeah.
Good. Then I will just share my screen. Let's see. Do you see the correct slide now? Not my notes?
It's your speaker screen, like with two slides.
Okay.
No, yeah, perfect.
Good. Okay, so my talk will be about modeling glioblastoma heterogeneity as a network of cell states. See if I can change the slide here. So I will just talk briefly about glioblastoma and some of the research questions that we had in this project. And then I will talk a bit about the data generation, since this is quite a unique data set, and then the modeling and the results part. So glioblastoma is the most malignant and also common of all brain tumors. And in Sweden, we have around 400 new cases every year. with the average age of the incidence being around 55 to 75 years old. And the prognosis for these patients is really bad. So they have a median survival of about 15 months from the time of diagnosis. And even though there are these rare cases of patients that fully recover, the overall 5-year survival is only around 5%. And if you want something to compare this with, so non-metastatic breast cancer has a 5-year survival of like 91%, I think. So it's a really, really bad disease to get. And the first line treatment against glioblastoma is surgical resection. That means that you remove as much of the tumor as you can. And then you follow up with something called the STAP protocol, which was established in 2005. And that is radiotherapy and chemotherapy with temozolomide. And this addition of chemotherapy was has been the biggest breakthrough in glioblastoma treatment for several decades. And that increased the survival from 12 to 15 months. So as you can see, there is this great lack of efficient therapeutic strategies to treat glioblastoma. And one reason behind this, or what we think is the reason, is that glioblastoma is a very heterogeneous disease. So it's hard to find one treatment that actually targets the whole complexity of this tumor. And that is why we want to study something called cell state dynamics here. So we know that glioblastoma is a highly heterogeneous disease, and it has this complex cellular composition, and this contributes to tumor progression, to therapy resistance, and in the end also to recurrence. And if you see this paper to the left, which was among the first to use single cell sequencing of glioblastoma cells. And here they actually conclude that glioblastoma tumors contain multiple cell states, and these have distinct transcriptional programs. And they say that there is this great need of a better understanding of the dynamics in order to establish good models and develop new therapeutic strategies. And then relating to this, we have this other paper to the right, where they do this exact thing. They study state transitions, but they do it in breast cancer. And they do it by sorting the cancer cells into 3 distinct phenotypic states based on surface markers. And then they follow them in cell culture over time. And what they can see is that these pure subpopulations repopulate the remaining states over time. And then they model their observations using just the Markov chain model, where they can see the cell transitions between these phenotypic states. So this project, we wanted to do kind of a combination of these two. So we want to use single cell sequencing to explore the states, since these are not at all as known in glioblastoma as they are in breast cancer. But to be able to observe these state transitions, we need some kind of time series data. And to model this, we thought of kind of a model like this, which is glioblastoma, where we have 2 phenotypic states, the go or grow, so migratory and proliferative states. And then cells can switch between these with some rate, but they can also die with some rate. And we thought that our model could be kind of like a data-driven generalization of this model, where we could get states from the single cell data and the transitions from the time series data, and then get functional annotation of each state, since we have the gene expression for each cell. So these questions that we started out with was, how can we go from a snapshot of current states, which is what we get from single cell data, to time series data? Can we define the states based on the expression data? And can we model how cells transition between these states? Do the states have different growth and death rates? Do the cells move freely, or is there a really clear hierarchy, which could then conform to the stem cell theory? And how are the transition and growth rates modulated by drugs? And here we see Erika and Rami, who has been doing all of the experimentation on this project. And the method that Erika developed to follow the cells over time makes use of a lentiviral-based barcode library, which has 100,000 unique barcodes and a BFP tag, which is like a fluorescent tag. So she transfects the cells at day -4, and she chooses these ratios to obtain 2,500 cells with a unique barcode at day 0. And then at day 0, the population is sorted using FACS to only keep the cells where the barcoding was successful. And thereafter, she followed them in culture for 21 days and did sampling after 7 and 14 days. And on these sampling days, she harvested 80% of the cells and sent them for single cell sequencing. And 20% were kept and seeded back for continued culturing. And then at day 21, all cells were sent for sequencing. So then we basically end up with a data set that looks like this. So for each cell, we have the experimental data or the expression data, but we also know what barcode it contained and at what time point it was sampled. And this kind of enables us to do lineage tracing of each cell to one of the 2,500 original cells. So we know like from where each cell came from. And first we just did some basic analysis to see that the barcoding was successful. So what you can see here is that on the x-axis there are all these individual barcodes, and on the y-axis you see how many cells had this barcode in them. So at day 0, the majority only of the barcodes were only in one cell, which kind of confirms that, or this was what we wanted to see, like the viral transfection was successful. And thereafter you can see that some of the barcodes expand very fast, while others grow slower or die out. And to look if there is actually like a biological explanation to this, we divided the barcodes into large and small clones and did differential expression. And there we could see that these small clones had an upregulation of this p53 pathway, which, well, it makes sense since p53 is a tumor suppressor. And the larger clones upregulated these SOX2 and stemness signatures, which also makes sense since these should have a higher proliferation rate. So based on these first analysis, we could conclude that the transcriptional profile and the clonal growth were correlated. And therefore we decided to move on and try to model these state transitions that I talked about in the beginning. So we defined this model that had 4 assumptions. And they were that each tumor cell exists in either of a certain number of states. And each of these states has a specific proliferation and death rate. The tumor cells stochastically transit between the states at some fixed average rate. And an externally applied drug can alter the transition rates, the growth rates, or both. And I've just tried to make a very simple illustration of this to the left here, where you can see a cell in a given state. For example, this green one, it can either divide and produce 2 daughter cells, it can die, or it can switch its phenotype and therefore end up in another state. And then we constrain these transition rates to be non-negative to estimate transitions in both directions. And then we approximate the system by simple linear first order rate equations, and we get this kind of solution. And so here, this xt and xt-1 is the state distribution over barcodes at time t and t-1. And this is what we get from the data. And what we are interested in is this A matrix, since it contains our transition and growth rates. And to solve this, we use, and E is the matrix exponential. And we use a first degree approximation of this to express just the sum of squares error, which we minimize using convex optimization to find A, given our experimental data. So now I went kind of fast, I think. Okay, yeah, I missed that one. This is our error. And if we do this, we can visualize the A matrix as a transition matrix, which kind of looks like this. So here you can see the transitions between states as a network. So each node is a state, which we defined from the single cell data. And we found that the data was best explained by 6 transcriptional states. And the arrows between the transitions, its thickness is proportional to the rate. So the rate of cells transitioning from state 1 to 4 is much higher than, for example, between state 1 and 3. And as you can see, we find this hierarchy in the network with a top state feeding cells downwards to intermediate states and finally to end up in kind of a sink state. And to see if this made sense, we have done a lot of functional annotation of these states, for example, just looking at the marker genes, using gene set enrichment analysis. And we actually see that this state 1 clearly resembles a stem-like state, and it makes sense that it's at the top of the hierarchy, while this state 6, for example, show a lot of senescent or apoptosis-like markers. So it would also make sense that cells end up there when they are dying. And then we find this invasive state and intermediate state, more differentiated state, etc. And from the A matrix, as I said, we can also derive the individual growth rates. And there you see that this state 6 has a negative growth rate, which means that the death rate was actually higher than the birth rate for the state. And this state 2 shows to be very rapidly proliferating, which also makes sense with the gene set markers we found there. So this was basically the first results we had. And it's very interesting to study the baseline network for glioblastoma, but we also wanted to investigate how drugs, for example, this standard of care drug, temozolomide, changed the dynamics of the cell states. And to do this, Erika designed a new experiment where she included 3 parallel arms, 2 treatments and 1 control. And to analyze this data, we just updated this A matrix to consist of a joint matrix, which is the same regardless of treatment arm, and the delta A, which is the change that each specific treatment has on the joint matrix. And then if we do this for temozolomide, we can plot this treatment, and we can see how this delta treatment, and we can see how temozolomide changed the baseline network. So here we see that it actually increases the transitions to state 5, which was an invasive state. And this is something that we also have found a lot of observations in the literature that temozolomide actually does, which was interesting since it kind of validates what we see here. So with this, I just want to finish and thank the people that worked with me on this project.
Thank you very much, Ida. Do we have questions on Ida's presentation? So are there plans to extend this model in some way? Maybe bringing in more states, or I guess it's very data dependent.
Yeah, so bringing in more states, I don't know, but we, first of all, we want to try to do experimental validation of the model. And you can do that in different ways. One is to do what they did for this breast cancer study, where you kind of try to sort them in pure states and then follow them over time and observe the transition rates. That is a bit hard because these states are not really, you have to have very clear surface proteins to be able to sort using FACS. And it's not all the states that has that. So we also looked at trying to do a CRISPR-based tagging system where you put the fluorescent marker in the genes that define the states and just using imaging, try to observe how they switch their states. But then we want to extend the project to, for example, look at how radiotherapy changes the transitions, and if there are patient differences in these networks, or if it looks kind of the same for different patients.
Okay, thank you.
Have you maybe, or have you thought about like some sort of sensitivity analysis that you find out which of the transitions might affect the system the most, or something like that?
Yeah, so we sent the manuscripts, which is now under review, and in there we have done exactly that. Like, so what is like the smallest perturbation we can do to the system to destabilize the growth, kind of. Yeah.
Okay, nice, thank you.
Maybe one bit more technical question. Since you approximated when you fit your model, you approximate an exponential with a linear function. Have you done any investigation how much, if you're losing much there, or like, I mean, since an exponential function can change in value very quickly, approximate by linear works, of course, as long as you're like, if you work with really small numbers, but could there be a downdraw to that approach?
So what we have looked at is just, we try to do like simulated data sets, and we see that there is, depending on like how many, for example, how many states and how many, the time series, like how long we follow the cells and how often we sample, it's not always that we are very precise. So there you have to design the experiment based on like that you have a certain interval in sampling, etc. So we have looked at that, but not more than that.
Okay, thank you. Any other questions? Okay, then thank you very much, Ida. And if there are more questions, as always, feel free to ask them on Zulip, or maybe even later in the speaker room, depending on if Ida will be there or not. Yes, then we can go to our next speaker, and that is Fredrik Ohlsson. And he's an associate professor of mathematics at Umeå University. And some of you might see him in the video now and think he looks very familiar. And that is because he actually used to be a guest lecturer at Mathematical Sciences in Gothenburg. So welcome, Fredrik.
Thank you. Let me just start by sharing my screen here. There we go. Can you see my presentation and also hear me? Excellent. Let me know if there are any technical problems during the talk. Thank you, Felix, for the introduction, and of course for the opportunity to present this talk today about symmetries in modeling in biology, and in particular in the example of model selection for the Hill model. I would like to start out by thinking a little bit about two fundamental questions in modeling in general, and of course in modeling in biology in particular. And the first one is, what is the biological mechanism governing the system? This is of course underlying every effort in modeling that we might make. And a related question from a sort of a different perspective is, starting from the data side, given measurement data on some system, what is the underlying mathematical structure of that data? And I will try to argue today that symmetries, in the context of this talk, they will be symmetries of differential equations, that they provide a framework to address to some extent both of these questions, and also understand a little bit about how they relate to each other. And they do this by providing a way to understand nonlinear properties of mathematical models, more specifically of their solutions, and conversely by extracting global properties of data that we might require our models to respect in order to be feasible models from an experimental point of view. The outline of my talk will be the following. So I will start by speaking a little bit about symmetries of ordinary differential equations in general. And then I will go on to describe work together with Johannes Borgqvist and Maria Svijovic in Gothenburg about applying this to the Hill model and to model selection among Hill models as a very simple example of the role that symmetries might play. But first, I want to say a few words about symmetries of differential equations. So the first thing is to sort of figure out how does the concept of symmetry apply to a differential equation. So if you think of the most sort of generic description of a symmetry is a transformation that acts on some object in a way that preserves the object or some structure of the object. So the most easily visualized example is consider a sphere in 3 dimensions. If you rotate this sphere through any angle around any axis, it will still look the same, assuming that you haven't made any marks on the surface of it. So this is the sort of prototypical image of a symmetry. And how can we apply this to differential equations? So in this talk, I will focus exclusively on a single first order ODE, but I will come back at the end and make some comment about how this can be generalized. So the first aspect here is to define what we mean by a transformation. What is the analog of rotating the geometrical object of the sphere? And in this case, we will use point transformations. So this is simply a transformation that to each starting point in the ty plane, where t is the independent variable and y is the dependent one, it will map each point in this plane to a new point. So this takes care of defining sort of the symmetry transformation. But of course, in order to be a symmetry, we need to ask that this preserves some structure concerning the ODE. And what we ask that this transformation preserves is the space of solutions. So if we start with a solution y here and act with the transformation gamma, this will take each point on the curve here and map it to a new point. And if gamma is a symmetry, then the resulting set of points here will define another solution y hat to the same differential equation. So this gives us the sort of the fundamental symmetry condition that tells us whether or not a particular transformation is a symmetry. As a very simple example, we can consider one of the easiest differential equations in existence, where the derivative of y is simply a constant k. Of course, we can solve this. We know the general solution. It's the form of a straight line with slope k. So this is then the meaning of the parameter k in the differential equation. And of course, we have some integration constant m here. Now I want to consider not a single transformation, but a family of point transformations parameterized by epsilon here, which take each point ty and maps it to the point where the time coordinate or the t coordinate is unchanged, but the y coordinate is shifted by this parameter. So this is the role of epsilon. And of course, if we insert these expressions for t hat and y hat, we easily find that this gives us a relation between the two that is also a solution to the original differential equation. So we have this symmetry condition fulfilled. And in terms of illustrating this geometrically, epsilon is then interpreted simply as the distance that we translate in the y direction. And gamma here is the family of transformations that takes each point on this original blue solution curve and maps it to the corresponding point of the transformed solution. So this is the basic symmetry concept. But in order to apply this to more complicated situations and more complicated differential equations, we need to put this in some geometrical context. And to do this, we can start by thinking about how can we define these transformations or families of point transformations in a more general way. And this can be done by starting with a vector field in the ty plane. And this can of course be thought of as just an ordinary two-component vector that points out some direction here. But we can also interpret this as a differential operator, where the t and the y are simply the basis elements in the tangent space. So this gives us sort of an operator interpretation of this vector as well. And using the exponential map, which we define simply in terms of its Taylor expansion, we can then look at what happens when we vary this parameter. And if we look at a single point, we will define a trajectory for the point like this, which is called the flow generated by the vector field, which is simply the integral curve of x. And using this, we obtain an operator that we can act on the original coordinates t and y, and obtain in this way the transformation. And doing this will give a one parameter set of transformations. Furthermore, these transformations will actually form what's called a one parameter Lie group of transformations. In particular, this has some nice properties, so that if we set epsilon to 0, you see, of course, that this corresponds to the trivial transformation that does not act on the plane at all, or has a trivial action. But we also see that we can invert any transformation simply by performing the transformation with the opposite sign of the transformation parameter. So in this way, we can define more general groups of parameters, or families of transformations, I should say. But we still have to make sure that they actually preserve the solutions to our differential equation. So in order to do that, we have to make sure that this family of transformations doesn't change the fact that solutions, that it doesn't change solutions into something that is not a solution. And this can be done by looking at the linear order in epsilon. So performing an expansion in this parameter, we can look at the linear order. And in order to not change anything here, we should make sure that the change in the left-hand side equals the change in the right-hand side. And the right-hand side changes in a fairly simple way. This is just a directional derivative of the function omega with respect to this direction defined by x. The left-hand side is slightly more complicated. It comes about because acting on the plane, of course, induces an action on the derivative y prime. And I don't want to go into detail about how this is derived, but simply state it to show that this gives us a linearized symmetry condition that we can now use. Because you see here that this is actually a differential equation where we have eta and xi, the components of the vector field appearing. So by making sure that we use solutions to these equations for the components, we are guaranteed that x generates this symmetry. So this is the technical background from a geometrical point of view. And now I want to talk a little bit about how to apply this in a very simple but concrete example. So now we will consider the Hill model, which I will not go into detail regarding how it is defined and why. As you all know, this can be used to model enzyme kinetics in a certain limit. But the important thing for this talk is that we can use this as an example, because removing all the dimensionful constants and just preserving the main mathematical structure, we obtain an expression like this, where the derivative of y, and y here we can interpret as the amount of substrate or concentration of substrate. We get a differential equation that relates the change in y to a function in t and y, as you see here. Of course, you notice that there is no explicit time dependence, but this is still of the right form for applying our machinery of symmetries to it. This number n here is called the order of the Hill model, or the Hill coefficient, and it can be interpreted as the number of active sites in the enzyme, although there is no need from a mathematical point of view to restrict this to integer values. So this is the model that we will now try to analyze a little bit using symmetries. And to do that, and in particular, looking ahead to the model selection problem, we would like to find a class of symmetries that distinguishes between the different values of this parameter, so that the structure corresponding to this symmetry should be different for different models that will allow us to make a choice between them. Starting from the linearized symmetry condition with this particular right-hand side omega, we can find precisely such a class. And I don't want you to think too much about the precise form of this, but the important thing to notice here is that we do have an appearance of this Hill order coefficient n in the actual generator. So that means that different values of n will generate different transformations, and it also means that if we look at the action of these, they will be generators, they will be particular to the precise model. So for n1 equals 1, for example, starting with the solution curve in blue here, we have one particular action that maps this to another curve. But if we change the value and look at n equals 2, we have a different transformation, and again for n equals 3. But all the time you see that this action on the plane, which sort of warps the ty plane, still maps the blue solution curve to another solution. So we still have a symmetry of the model. And this can now be used to define and perform structural model selection. And the idea is here that starting with a number of candidate models and some symmetries that are associated to each model, we can perform the following 4 steps in order to try to fit this to some given experimental data. The first thing that we do is that we apply, for each model, we apply the corresponding symmetry transformation gamma. That takes our original data and produces some transformed data set. And to this, we can then fit the model in question. And this can be done in many different ways. We have used ordinary least square fitting here. The important thing is that before we evaluate the quality of this fit, we perform an inverse transform on the model. So we take the model, which is represented here by the red line, and we transform it back to the original parametrization of the ty space. And then we compare it to the original data to see what is our quality of fit, and we measure it here using just the ordinary RMS value in which we call rho here. And importantly, this is now a function of epsilon, because depending on what the value of epsilon we started here, we will move further and further away from the trivial transformation at epsilon equals 0. And the rationale behind doing this is that if this gamma epsilon is indeed a symmetry that is realized in the data, then we expect that the model and the data should transform similarly. So it shouldn't matter how far away from 0 we perform this transformation. Whereas if the symmetry is not realized in the data, so the model is not structurally compatible with the data, it will distort the data compared to the model and reduce the quality of fit as we increase the parameter. And to exemplify this, we have set up a very, very simple test scenario where we consider 3 Hill models as candidates. So we use simple integer values for these fitted models. And we generate simulated data also from a Hill model. So this is a very simple scenario. But what we can see, for example, if we look at the simulated data with n equals 2, we see that we get some, if we repeat the simulation and fitting procedure, we get some results like this. So of course, the average gives us the correct result here. The average selects the n equals 2 model if we look at epsilon equals 0 now. So this means doing just ordinary least square fitting of the models. But you see that if we don't have a lot of iterations of this, there are still significant uncertainties here. And because of these overlaps of the confidence intervals, at least in this case at the level of 1 standard error, we don't actually resolve these sufficiently. But on the other hand, if we start increasing the parameter epsilon, we now see that as expected, the model with the correct structure, which is in red here, n equals 2, it has approximately constant error when we increase epsilon. Whereas the other 2 models, they start to diverge, meaning that this overlap of confidence intervals is resolved and we can reject these models as not structurally compatible with the data. As I said, this provides a very, very simple example of the use of symmetry methods, because they have a much wider range of application throughout modeling in biology. It can be used in systems of ODEs, systems on partial differential equations, and we can take both of these perspectives that I've been talking about during this presentation, that we can try to infer some symmetry that we expect from a biological point of view and make sure that that is manifest, or we can look for structures in our data and try to make sure that we create models that respect those. And more generally, I hope that going back to the discussion after Gunnar's talk this morning, that I can convince you that this gives us a way to use differential geometry, at least in some way, to understand mechanisms governing biological systems by using it in modeling. So with that, I would like to thank you for your attention and thank, of course, my collaborators, Johannes and Maria, again, and I will happily take questions from the audience.
Thank you very much, Fredrik. Do we have questions?
I was wondering, when you define these generators, I think these gammas, the first time, you talk about that they have to fulfill the symmetry, and you define that in a linearized sense. Does that mean that these generators only work up to first order, or is there, do you actually have a proof that the higher orders are irrelevant?
Excellent question. This has to do with the fact that they generate a Lie group, and the Lie group is completely specified by what happens at the first order. So there is no linear approximation at work here. This gives you the absolutely correct transformation regardless of the value of epsilon. Although I should say that this is typically called a local Lie group because there is some limit. It might be that you have disconnected components of your group, for example, so that you can't reach from epsilon equals 0 to precisely every element, but it's certainly not only valid in the linear regime.
Okay, thank you.
I also have two technical, possibly related questions, and apologies if I'm a bit rusty with these concepts. I'm curious if you could, instead of an invertible differential operator, could you have something like a Galois connection with another space? So not fully invertible, but, and still reasonably conserve some symmetry, depending on the model and system.
That is an interesting question that I don't really have a good answer to. I mean, typically, because of the way that symmetries behave. I mean, if you look at them as transformations, and don't worry too much about this vector field perspective, they will be naturally elements in a Lie group. And then you have this structure because, of course, there's a group structure that symmetries can be multiplied or combined. You can perform successive symmetry transformations. And that means that you have this natural Lie group structure if you think of them both as the group and the manifold. So I'm not sure how to answer that question. At least I don't know what the appropriate answer is. But it's certainly interesting to consider cases like that where you put some additional, or rather where you remove some of the restrictions that we use here.
Interesting, because the motivation was related to my second question. If you were, so I understand this is for ODEs, but if you for some reason had a discrete space that you want to map to, and again, within reason, conserve some symmetry, and I'm not sure that you would go about it by a Galois connection or something similar, but how would that go?
I mean, there are certainly ways to do this with discrete symmetries in general and in biology in particular, I think are very interested. Of course, in our application here to the model selection scenario, that might be difficult to reconcile because we are sort of limited to doing things for reasonably small epsilon. Otherwise, the effect of any measurement error can at some point obscure the actual structure. So it's not trivial to generalize this to discrete symmetries, but they are very interesting because, of course, the same machinery works for discrete symmetries. It's just that then you don't have these one parameter Lie groups or more general Lie groups anymore.
Right. Okay. Very interesting. Thank you.
Thanks.
So yeah, thank you very much, Fredrik. If there are more questions, as always, Sulip or GatherTown. We will take a 10-minute break now, and then let me just double check so I'm not saying anything wrong, at 11:10 we will start up again. And thank you very much to our speakers of this second block of today, Sebastian, Ida, and Fredrik. And see you soon in about 10 minutes.
And I think there was an initiative for that people spend the break together at GatherTown. So if you feel like that, please just enter the room that you find at the social event.
All right, welcome back for the last session of talks for today. And we'll start off this session with Peter Jagisch, who is a professor emeritus at the Mathematical Sciences Department here in Gothenburg. And he's had a long and fruitful career in graph theory, in particular in the area of branching processes. And we're happy to have you here today, Peter.
Thank you very much for those nice words. And I shall talk about a very old problem and a very classical problem and about a very classical mistake, where I will claim that the people who did the mistake were basically right. They asserted that almost all, or actually all, essentially all populations must die out. And what they really said is what you can see on the slide here, that, I think I'll do it like this, that all surnames, families, and populations tend to extinction. And they added an argument. This result might have been anticipated, for a surname lost can never be recorded. Now, you may, hope it doesn't. Oh.
Sometimes you have to click on the presentation once and then you can use your keys.
Pardon, where should I click?
Just
on the
somewhere on the presentation and then you should be able to use your keys.
Yeah. This problem was actually first formulated by a French probabilist, but Irene Jules Bien-Aimé, but his contribution was forgotten. And the person who, where the whole theory started was Sir Francis Galton. Galton was a very strange fellow. He was one of the great savants of the 19th century. He was actually Darwin's cousin. He was an explorer, an anthropologist, a geneticist, a psychologist. He was the guy who discovered the concept of correlation. And he was also a very practical person. He discovered fingerprints and how they could be used in criminology. But he was also a slightly doubtful person. He was the person who introduced the concept of racial hygiene and the ideas of measuring the head to find out which races are the most intelligent. And it's quite clear that such a person should be worried about the decline of noble families. Where are all the Tudors, Stuarts, and other noble families of Britain? He was quite a complicated guy, but he was also a child of his time. In 1873, he formulated the problem the following way. A large nation, of whom we will only concern ourselves with the adult males. You can see what kind of a guy he is. He talks about human population, where the reproducing unit are the females, but still he concentrates upon the males because he's interested, or he pretends that he is interested in family names, which in his mind are always inherited from father to son, where the adult males, who each bears separate surnames, colonize a district. And then he says, their law of population is such that in each generation, a certain fraction have no male children, another fraction has one child, two children, etc. Find out the, well, let's call it the probability of extinction to make things easy. He couldn't solve the problem himself. He turned to the brightest person of his class at Oxford, who was a clergyman, Henry Watson, and Watson solved it. He solved it in the sense that he formulated what we now know as a Galton-Watson or simple branching process. The structure here is indeed very simple, but it catches something essential of population growth and decrease. So individuals, all individuals are independent of each other, except of course that you must be born by your mother, but they reproduce independently, and they reproduce according to the same probabilities. Probability P0 to have 0 children, P1 to have 1 child, and generally Pk to get K children. So this is an asexual reproduction. We would say that we only consider the females. Galton, of course, would say that he would only consider the males. The sequence of population sizes is called the Galton-Watson, or sometimes a Bien-Aimé-Galton-Watson, or simple branching process. If we introduce the reproduction generating function, the sum over the probabilities of having K children multiplied by, well, S to the power of K, S is the argument of the function, then it is easy to show that the extinction probability Q is a fixed point of this reproduction generating function. It is also easy to see, since probabilities always sum to 1, that f of 1 equals 1. Hence, said Galton and Watson, Q must be 1. It's interesting that such intelligent people, and at least one of them very well versed in mathematics, could make such a simple mistake, because there can be other roots. They didn't think of those. And even stranger is that during 50 years, Galton's and Watson's claim was not disproved, though of course it was criticized. But today, of course, we know it's a textbook fact that if the mean reproduction, which in population dynamics we usually call M, epidemiologists, they love calling it R0, as we now all know. This is the famous reproduction number. If that one is bigger than 1, there are actually 2 possible roots. And one is, the number 1 is one of those, but the real root is always the smallest non-negative root of the equation f of x equals x. And this root is smaller than 1 precisely when m or R0 is bigger than 1, giving an option that the population will not die out, but increase exponentially. And this dichotomy, which was proved much, much later, more than 50 years later, 52 years later actually, is basically true not only for Galton-Watson processes, but also much more generally in continuous time and in discrete time. And for populations which can give birth repeatedly or just once, they can be one type or multi-type, but there you have to be a little bit more careful. And the theorem is basically true as long as individuals reproduce independently of each other and with stable, unchanging reproduction probabilities. But what happens if the population interacts with its environment? This is the question that we shall discuss during this short talk. So then we have to approach if we can handle interaction with, it's easy to say interaction with the environment, and there is a feedback loop between the population and the environment, etc., etc. But this is merely talk. We have to have a clear and lucid and still general pattern for population size dependent. So we shall assume that there is a large number, which is the carrying capacity of the population considered. This wording suggests that the population turns subcritical above K. But what does that mean precisely? It means that, well, let's now assume that the population changes. We shall have a very general model. We shall do no assumptions whatsoever about independence between individuals and no assumptions on the individual levels. We just assume that the population changes size at discrete time points in underlying time, which can be continuous or discrete. So the population has a starting size, which we will denote by Z sub 0. Then there is a first size change, a second size change, etc. And we define the population size at the nth, after the nth change, as the size at the after the n minus first change plus the change that occurred right now. And in this, the change variables, they are not assumed to be IID or anything like that. We assume only what I say here now. Either the change is negative, then of course it cannot be, and we can never lose more than the whole population. Or the change is strictly positive. But in a non-extinct population, a change is always a change. That is a very crucial assumption. In an extinct population, there are no changes. Then you can think of these changes in multifarious ways. You can think of this being individuals either dying or reproducing one by one, but this is not crucial. They can die in heaps or they can reproduce together several individuals as long as the special restrictions I have made are kept. So let now tau sub n denote the time of the nth population change, if there is any. If there is no one, we write tau sub n to equal infinity. And a sub n, this is summarized, is the sigma algebra of all events that occurred up to that time. And we can have really all events, not only concerning the changes, but say that this individual did that, or that individual, well, turned out to be a cancer cell, or something like that. Well, there could be many, many things occurring. But this is the sigma algebra of everything that has occurred up to time tau sub n. And now I give a strict mathematical formulation of our assumptions. The expected size of the population at time tau n plus 1, at the next change, given the sigma algebra of all events up to the nth change, it is smaller than the present size, provided the present size is bigger than the carrying capacity. That is actually the definition of carrying capacity. On the mean, the process tends to decrease when it is larger than the carrying capacity, but only on the mean. And then the second assumption is the one I already mentioned a couple of times, that if the process is extinct, then it is extinct. And then the third and crucial assumption is that there is a uniform risk for decrease of the population, provided that the population is small, that is, the size is under the carrying capacity. And so we come to the main result. In words, we have assumed that the population tends, or rather is expected to tend to decrease when it is too large, and that if it has died out, it will not resurrect, no immigration or anything like that. And finally, that there is always a risk of an individual dying. Very reasonable. Those are the only assumptions. I stress that. This is a collective approach. We have said nothing about individuals, in contrast to the individual-based models like those in birth and death, in the birth and death tradition, or in branching processes. So in that sense, it is a little bit more similar to the deterministic differential equations-based population dynamic approach. It covers an extreme range of individual behaviors, and unlike the differential equations modeling of population dynamics, which is also, as I said, collective. It makes no hidden assumptions about, say, lifespan distributions or independence between individuals. This is very often not realized, that when people assume differential equations governing the development of epidemics or populations, they are implicitly assuming exponential distributions of lots of things, which is very, very unbiological. Anyhow, if we make the simple assumptions that I mentioned, then the population must die out. The probability that Zn tends to 0 equals 1. I shall very quickly sketch the proof idea. I don't have much time left, but the basic argument is simple, and it uses martingale theory. When the population size is above k, the population size sequence behaves like a super martingale. A super martingale, a non-negative super martingale, must converge. Downwards jumps in this very broad model have size at least 1. So convergence must be to something below k. Because above k we always can, well, there is always a difference of 1 between two subsequent states. And whenever we are below k, then there is a probability of a direct path to extinction, namely that k times in a row, capital K times in a row, the misfortune occurs that we move downwards. Thus, sooner or later, the population must die out. This can, of course, also be expressed with the help. There are, of course, lots of technicalities buried, but this is the general argument. I shall not bother you with these technicalities, but it works using stopping times, like the first time you are below k, first time you are above k after you have first been below k, second time you are below k after you have been above k, etc. And then you work with these numbers, and I shall, as I said, not go into detail. The result is that what I have said, and we can conclude that the probability that the population dies out equals 1. Those who are interested in the mathematical argument, which I have so brutally sketched here on this brown slide, can have a look instead at our paper. This is a joint paper between Sergei Zuyev at our department and myself, and it is published in the very most recent issue of the Journal of Mathematical Biology, where it is actually the very first paper, and there you can see the simple argument that leads to the complete result. And the complete result is that in the long run, we are all dead. Thank you very much for your attention.
Thank you very much, Peter. Do we have a quick question on this talk? Otherwise, it's almost time to go to the next talk. All right, then thank you very much, Peter, for joining the workshop.
Thank you very much. I shall remain with you.
Great, awesome. Then we're coming up to our last speaker for today, and that is Jeanette Hellgren Kotaleski, who is a professor in neuroinformatics at the Department of Computational Science and Technology at KTH in Stockholm. And her research is focused all around the brain. It's focused on understanding neuromechanisms, and the goal is to learn about how the brain processes information, how rhythms are generated, and also how learning in motor systems takes place. So welcome, Jeanette.
Thank you for having me here. It has been an interesting 2 days. And now I will switch to modeling of the brain. So I will try to illustrate how we can use data-driven models and mechanistic models to better understand aspects of how the brain works. And of course, this is just one of all possible model philosophies applied to the brain in the field of computational neuroscience. So, but this will be illustrate one approach. So first, a few facts about the brain. As you probably know, it has 10 raised to 11 neurons and same order of non-neural cells, the glia cells. Every neuron connects to other neurons with several thousand synapses on average. And this, if we just consider that a synapse is either on or off, then the brain has this number of states, but in reality it's much more because all the individual neurons also change all the time and synaptic properties also. So it's a lot of states in the brain that can switch every moment, and still it is very energy efficient. It runs on less energy than old-fashioned light bulb. It can last a lifetime without spare parts. And I mean, we all agree that everything we know about the world, everything we interfere with the environment, this is because of our brains or the tools that our brains have created. So one reason why we want to understand the brain is, of course, due to curiosity. This is a fascinating system. But of course, also, if we understand the brain, we might be able to extract new principles for computing and AI and robotics and so on. And maybe the most pressing need is that we want to counteract diseases. And as you might, brain diseases, and as you might know, the healthcare cost in the Western world, it's a third of that relates to brain-related diseases, and this is growing when we get older. So it's actually very expensive to not understand the brain better, and of course, not to say about all the suffering that people go through. So this is a quotation that you might have seen before. It's denoted to Sidney Brenner, and he said that progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. And this is probably true for many fields, but when it comes to brain science, this is really a very good description. I mean, we have over the decades more and more rapidly developed new techniques to measure different things in the brain, from the molecular subcellular level up to the whole brain level, both static and dynamic data of different sorts. And also we have computational capabilities. We have supercomputers. We can store the data, exchange the data, analyze the data. So in this way, we generate a lot of new insights every day on how these subcellular, cellular, and network phenomena correlate with the healthy and diseased brain. But the challenge is, of course, that a typical lab is focusing on one of these boxes. And this also means that this excess of facts is not necessarily integrated that well into knowledge and understanding of what one can call the causal chain of events. And what this means is, for instance, over multiple scales. So we all know that a population of active neurons correlate with behavior and can predict behavior. And the properties of these neural networks is, of course, controlled by what happens inside the cells every moment. And also we have all these dynamic and complex processes. So I guess we all agree that we need to use modeling and simulations also to integrate data for the brain. And then I should just point out that even if we have a lot of data, it still represents really sparse data, because if we want to build a network model of a piece of brain tissue, then we haven't measured all cells and all the synapses, obviously. So we have to start with sparse data and develop modeling workflows or pipelines and tools supporting those so that we can actually predict missing data and come up with a dense model and generalize. And this is, if this is done in a standardized way, so it can be autonomized, then this is also something that really facilitate collaborations, because if we want to model large part of the brains, it has to be collaborations between different labs, and models have to be reusable and repeatable in different labs. And this also will assume specific types of data that I will illustrate. So in my group in Stockholm, and actually in many other groups in Stockholm, also experimental groups, we are interested in basal ganglia. So the basal ganglia is a forebrain structure that is involved in behavioral selection of behavior, decision making, reward learning, motivation, and so on. And if things goes wrong, you can get diseases such as Parkinson's disease, but also other neuropsychiatric disorders are involving basal ganglia, such as ADHD, schizophrenia, and so on. So this is a basal ganglia. It's this forebrain structure that I said, and the input stage of the basal ganglia is called stratum. It receives input from the whole cortex and also from thalamus, and then this information is channeled through something called the direct pathway and the indirect pathway to output stages of the basal ganglia and motor centers in the brainstem, but also there is a loop back through thalamus. So we also have this cortico-basal ganglia thalamic loop that in turn influence planning, and motivation and decision making. So now we'll start with illustrating how you can build a model of this input stage of the basal ganglia that in the mouse consists of 2 million neurons. So what would be needed? You would need to know type of neurons, how the neurons look, morphology, the electrophysiological behavior of the neurons, expression of ion channels and so on, how many cells there are, how cells are connected, and how these synapses behave. And then the workflow could be the following. First we have to have experimental data of all these things, or to sufficient amount at least. And then we create sort of a database of instances of the individual neurons, as many as we can, and for the different neuron types. And then we place the neurons in the volume of the stratum in this case, and connect them and simulate them. So let's start with the first steps, where we try to collect and build models of the individual neurons. So different experimental approaches, including the full transcriptomics, have shown that in the stratum you have 95% of all cell types are spiny projection neurons, which give rise to this direct and indirect pathway. So they have their cell bodies in the stratum, and then they project out from the stratum. And then there is 5% of interneurons in the stratum, and these interneurons, they come in different types, with different types. Most of them are inhibitory, GABAergic. And then we have reconstructions of most of these neuron types. This is just an example of an interneuron called LTS. This is a dendrites cell body and the dendrites, and this is then the axons that connect to other neurons. So this kind of data we can get from public databases, but also, of course, from collaborations with experimentalists in Sweden or abroad. So then we have the morphology of various neuron types, and also we assume we have the electrophysiological properties of these different cell types. And then the question is how can we reproduce that in the model? And then we have to know which ion channels we have for a particular neuron type. And as I said, we have the full transcriptomics of the stratum, fortunately. So here we have a good guide what type of ion channels could give rise to the electrophysiological properties for different neuron types. And also there is already a lot of models out there for the individual ion channels. So it means that we model the neuron and put channels onto the cell surface, sort of membrane surface, and then try to reproduce its electrophysiological behavior. And this could need hundreds of ordinary differential equations per neuron, or even 1,000. So just a few words about this thing that the neurons have a complex morphology. So how to deal with the spatial problem. And if this is a piece of a dendrite, then the approach is typically to chop it up and represent it as compartments, where every compartment is then modeled as an equivalent electrical circuit that could look like this. That in this small piece of the membrane, you represent passive properties, but also these active ion channels, the voltage-gated action channels that are typically modeled using a Hodgkin-Huxley mechanism or more advanced methods. And then it means that you can track the voltage deflection over the membrane, over the whole surface of the modeled neuron. But still it used to be a very frustrating, and still is a frustrating challenge to try to fit the model with the data, because you have to optimize the density of ion channels and balance them against each other. But today we have help from computers. So this shows a snapshot of such a process, where the challenge is to have the model, the blue curve, resemble the data more and more. And this is then repeated with genetic algorithm. This is a snapshot. And this could be the end result of a particular neuron type, in this case, one of the spiny projection neurons, experiments modeled for various positive and negative current injections in the soma. And as said, we also need to model several instances of the same neuron type. This just shows the voltage currents relationship and the spike frequency current relationship of 4 other neurons. And as you see, there is a big variability also between the same type of neurons. One can also capture more subtle effects. So when a neuron is active and it spikes, then you have a big depolarization in the soma, and then this depolarization spreads backward into the dendrites. And since we have voltage activated channels, calcium channels in the dendrites, we also see a gradient of calcium along the dendrites. So these things can be captured. And then we assume we have a database of neuron models for the different cell types. So then it's time to place the neurons into the volume of the stratum and build the network. So then what type of data do we use for that? Well, first of all, we have to know the volume or how the stratum looks. So we can use like the mouse brain atlas at the Allen Brain Institute. And then, so then we know the structure, and then we can place neurons there. And also over the years, a lot of pairwise recordings between neurons or tracing studies and so on have helped to figure out the connectivity of how different neurons are connected. So, but we still have sparse data, as said. So this is how it could look. This is the stratum on the left and right side. And if we look at the small portion there, it would be quite crowded with neurons. And if we add on the dendrites, it's even more crowded. And then the question is, how do we set up the network based on this? Because now we have filled the volume with the right density of neurons, but how do we set up the network based on this? And here we can use sort of touch detection. It means that we look where is a dendrite and an axon close. If an axon and a dendrite is not close, then it can't be a contact synapse. But if they are close, it could be a synapse. But most of the cases it's not. So first we see where it could potentially be a synapse, and then we apply pruning rules. So these pruning rules, this is principles for how neurons are coupled, and this information has been collected from many parts of the brain. So one rule can be that a particular neuron type project to distal dendrites of the postsynaptic neurons, and others could project more close to the soma, and some are randomly distributed, and so on. So these kind of pruning rules we then use when we do this touch detection. And then we actually have a dense network, a full-scale network of, in this case, the mouse stratum. Because we have all the cells and we have predicted all the connectivities. And that means, for instance, that we have then predictions on how connectivity between two different cell types decay over distance, or if two cells are coupled, with how many synapse connection points are they coupled, how many presynaptic neurons do you have a particular postsynaptic neuron type, and so on. So then we also have to look at to adjust the synapses, the connectivity, the characteristics of those. And this is a typical experimental paradigm when you look at synaptic dynamics. So you stimulate the presynaptic cells fast, and this is a postsynaptic response. And in this particular case, the response is going down, and then this is called synaptic depression. And then you wait for a while to see if it has recovered. And we also have the opposite behavior. We could have facilitation and so on. And so this we also match. So the red thing is the data, and the black thing is models. So then we actually have a full network. This is sort of a proof of concept. So then we have like 2 million neurons, but usually we don't simulate this amount of neurons because of then we have to have even more powerful computers than we have at the moment. And so this is sort of a proof of concept, and we can do these raster plots, look at the individual neurons and so on. And most important is then that we have created this quite detailed in silico sort of platform, which we can use to do in silico experiments that are difficult to do in real life. So we can perform specific lesion experiments to better understand, for instance, the role of the different neuron types, or we can perturb model parameters to see which effect that have on the network activity. We can simulate neuromodulation, for instance, investigate how this striatal network in this case would respond to inputs with different statistics, degrees of synchronization, and so on, which has been measured in the brain in different ways. And most importantly, also we can try to explore the hypothesized function of the striatum to see if that can be actually implemented into these detailed networks. So that was that was sort of a system level model of the striatum. But when we have reconstructed these detailed neurons, one can also actually use them to understand better, for instance, how a neuron like this integrate synaptic inputs from the surrounding. So this is an example of that. This is then the model of the neuron. This is a soma. And then we look at, as a test case, what happens if we have synaptic input quite proximal to the soma or further out in the dendrite. And here we simulate a phenomena that is seen not only in the principal neurons in the striatum, but actually in cortex and in many parts of the brain. So if we give like 10, 20 synaptic inputs to the soma region or close to the soma, then of course the cell is depolarized, but then when the input disappears, it goes back to the resting state. But if we do the same thing in the distal dendrite, so here we have given the same synaptic input in the distal dendrite, then we can activate NMDA-dependent responses. So when we remove the input, this depolarization stays on for many tens of milliseconds. And that also means that while we have this so-called dendritic plateau potential, there is also depolarization spreading throughout the neuron, which means that if we get this input to the dendrite that elicits this plateau, the cell goes up in a state where it's more ready to listen and respond to additional inputs that come within the nearest tens of milliseconds. So this means it's sort of a short-term cellular memory or attention. So dendritic plateaus facilitate then the spatial and temporal integration of excitatory inputs. This is also quite intuitive. And also it's generally known that these plateaus also facilitate synaptic plasticity, because a lot of calcium comes in, and this is the prerequisite for different types of plasticity. So it's obviously very important how these plateaus are regulated. And that's also why it is important to, for instance, ask, so what is this, what are these interneurons, inhibitory interneurons, doing to control the plateaus, or can they control the plateaus? So this is also simulation experiments that one can do. So if this is a postsynaptic spinal correction neuron, then these inhibitory interneurons, they can either sometimes sort of inhibit the spine correction neuron close to the soma or proximal parts, and some are rather inhibiting them further out in the dendrite, and also with different dynamics. And then if one do simulation experiments on this system, it is generally known that proximal inputs are typically most efficient to prevent spiking in the soma when the depolarization is because of summation of depolarization from a lot of different dendrites. But if we have evoked a plateau potential in a specific dendrite, then you can still inhibit the soma, but this plateau potential is not killed. So when you remove the inhibition, the depolarization will jump up again. This is in contrast to if you have inhibition in the distal dendrite close to where you have this plateau potential, then you can kill the plateau. And this really means that you change how much this cell can listen to what comes next during several tens of milliseconds. So this also sheds light on what is the role of this distal inhibition, that if you do pairwise recording, you hardly notice any effect. But if you assume that you have these active dendritic properties, then this distal inhibitory input could be very, very, very efficient. So what I have said so far is that, or try to exemplify, is that it's possible to reconstruct the striatal microcircuit in a data-driven manner, at least in the mouse. But in principle, the same workflow can be used in any species when you have enough data. Also in this, we can elicit these dendritic plateau potentials, which then facilitate significantly the temporal and spatial integration of excitatory inputs in the following tens of milliseconds. And also we have shown that cell type specific inhibition of these dendritic plateaus is possible. And what is quite interesting is that even though we have thousands of excitatory and inhibitory connections onto these spine correction neurons, or principal neurons in general in the brain, it could be enough that a few distal inhibitory synapses can actually influence and kill these nonlinear dendritic plateaus if they're timed and localized optimally. And actually we are playing around with that right now to investigate how input patterns can be sort of recognized and stored in the synapses and how these patterns can be learned because of this nonlinear property. So we are actually trying to see if a detailed cell of the stratum can learn this XOR problem, which is known to be a nonlinear problem, and it can. So this is sort of interesting. So it really highlights that a particular neuron is really a computational machine. It's not just something that summates input. It has more to it than that. So now I also should say a few words at the subcellular level and link it a little bit to systems biology. So if we know that we have these elaborated morphologies of neurons, and that it is important for how to integrate synaptic input. And if we look onto these dendrites more closely, many have these dendritic spines, which is where excitatory synapses and also sometimes inhibitory synapses are sitting. And these spines are also important for plastic changes and learning. And to understand then how network activity shapes plasticity and learning in the brain, of course, we have to go down to the subcellular level and understand better some of these receptor-induced cascades. And then many labs have over the decades tried to understand what kind of receptor-induced cascades or what effectors are important to induce strengthening of the synapse, LTP, or weakening of the synapse, LTD. And these cascades, our group, but also others have modeled. And then a typical approach is just to represent these reactions using bimolecular reactions, where you have to know these reaction rates, forward and backward reaction rates. So typically we use ODEs, but sometimes if one thinks that the spatial constraints are important, it could also be stochastic events, because a small dendritic spine is actually very, very small, and some of the molecules are there in very small amounts. So actually a deterministic approach would not really capture what happens. So what kind of questions do we address with this in the brain? This is an example. One thing that we have tried to address was why is it so that in reward learning, so in stratum was involved in reward learning. So why is it so that in reward learning, action has to come before the reward? Otherwise, an animal or won't learn at all. The reward can't come just before the action, and it can't actually be combined either. It has to come a little bit afterwards. And some years ago, it was shown that this timing requirement is actually, it has a correlate at the dendritic level and actually at the dendritic spine level. They could see that if they uncaged like dopamine and calcium and so on, it strengthened the synapse if it was timed in the right manner. And this was interesting. So here we have tried to model the reward, dopamine-induced reward cascade, and also represented aspects of what the action leads to. So the action is induced by inputs from cortex, and then there is a lot of calcium coming into these neurons, and so it's well known what cascades are necessary. And then we try to look at cross-talking proteins to see if we could find some proteins that is known to be here, and which could implement this kind of timing dependency. So this is questions that we can address. Another example is to try to model many of these pathways and then activate the system with in the same way as they do for experimental paradigms when they induce long-term potentiation or long-term depression, and tune these models to reproduce that. And then we used it to ask, because the stratum is also involved in drug addiction, like alcohol addiction and so on. So when you have used alcohol for a long time, then of course these things changes. And also when you withdraw, it seems that some of these changes are not reversible. And if we introduce those in this network and repeat these activation patterns, then we see that the balance for the system to shift between LTP and LTD is shifted. And this could potentially also explain relapse, why it is so easy to go back, even if you have stopped taking drugs for a long time. Then the third example.
Sorry, Jeanette, sorry to interrupt you. Is it okay if you wrap up soon?
Yes,
because we're running out of time.
I just have a few slides.
Okay.
So this is the third thing that we have also addressed, the detection of transient neuromodulatory inputs. So in the brain, I mean, it's really these sub-second neuromodulatory inputs that could be important to detect. And in this particular case of the corticostriatal synapse, one important motive is for long-term potentiation is cyclic AMP-PKA cascade. And then we model this quite simple system, but actually it turns out that depending on how you set the parameters, you can either detect these kind of transient changes or not, because the dynamics really play a role, and this cannot really be measured today, maybe tomorrow, but not today. And then we have collaborated also with molecular simulation groups that try to understand better how the facilitatory and inhibitory G proteins can interfere with, in this case, adenylyl cyclase type 5 that is responsible for the cyclic AMP cascade in these neurons. So questions like how fast do the reactions occur? Can the ternary complex form? Can both G protein bind at the same time? And what is the activity of this trinary complex? And then the molecular modeling can do a lot of predictions, like compare AC5 to other AC isoforms, or predict the binding of these G proteins, investigate how the inhibitory G protein inhibits AC5 and so on. And to make a long story short, it turned out that the ternary complex is probably stable, and it is inhibitory, so it's inactive. And this actually gives rise to a very large synergy in the detection of these coincident transients that are already known to occur in the stratum. And this is quite interesting that this is for the AC5 subtype. It might not be the same for other AC subtypes in the brain. So just to conclude that, of course, it's very difficult to constrain these subcellular models, but in addition to optimization workflow that we have heard about, also one can start to use molecular approaches to constrain the parameters. So I have mainly talked about the cellular level modeling and try to highlight that you can build microcircuits of that. But of course, I also wanted to point out that the subcellular, to combine this with systems biology approaches will be extremely important, because a very key feature of the brain is that it is learning. It's an amazing learning machine. It changes all the time, adapts, and so on. And for some of these models, you can also constrain it with molecular level simulations. Something I haven't mentioned at all is that if you have a model of for instance, a motor system in the brain, such as the basal ganglia and so on, you can also try to model that in closed loop with the environment. I just have one slide on that, illustrates something we did several years ago. Then we had a very simplified, it wasn't at all a data-driven, it was sort of top-down, hypothesis-driven, very simplified model of the basal ganglia coupled to a simplified model of the brainstem and spinal cord. And then we can see if it's possible that some neural principle can actually control how, in this case, a lamprey is swimming and trying to swim towards these things and hit them. So this is also, I hope you get the, I don't have time to go into this, but I hope you get the idea that if you are studying motor systems, then you can also interpret and look at how neuron population can control behavior. So I just wanted to acknowledge that a lot of people are involved in this, a lot of experimentalists at the Karolinska Institute, and also modelers at the cellular, subcellular level, and I said also these people that can do molecular simulations to predict and constrain these systems biology models. Yeah.
Okay, thank you very much, Jeanette. Now it's a little bit over time, but maybe if we have one very quick question that we could definitely fit, if there's anything directly.
Maybe just a quick technical question. I'm sorry, just a technical question. It was a very nice presentation. Thank you. For you mentioned data size is a problem. Does your framework smooth that? Does it use GPU acceleration? Have you looked into that?
Yeah. So actually, at the different scales, we have particular simulation tools, simulation software, and many of these tools have now been optimized to supercomputers, including GPUs and so on. And especially this simulator engine that we use for the cellular level, that is Neuron. I mean, there exist others as well, but we use Neuron, and there they have enhanced it. It's called core neurons, and there is also attempts to actually implement it on GPUs. And actually, as we speak, we are collaborating with a group that is actually trying to do that. Because then we can even simulate not only the advanced morphology, but even these spines. So to have too many compartments when we have large simulations, this is quite demanding.
But it sounds fun. Thank you.
Great. Another question came in from Gunnar. If you, Jeanette, connected what you talked about to the many other functions of the brain and neural cells, like the metabolism, the control of blood flow, these things?
We have just looked at neurons but I mean, others, if you have, you can, there are other tools. So if you have these detailed cell models, you can, for instance, synthesize like the local field potentials and fMRI and things like that in a sort of bottom up and try to predict it. And also we have this, one can also plug in these models, or actually maybe not these models, they have to be reduced, but anyway, spiking point neural models, or even neural mass models that when you have sort of population of those into a simulation framework called the virtual brain. I don't know if you've heard about that, and there you can also try to predict this kind of like fMRI signals and so on. But I mean, that's of course very interesting, especially when it comes to model human data, because in human data you just have like that level and maybe some gene levels, but you have very little on the level in between when it comes to cells and spiking cells. So this is extremely important, actually. Yeah.
Yeah. Thank you. Sorry, Gunnar, you can ask in a second. Just wanted to wrap up, and then I will leave the Zoom session open so you can keep discussing. Just wanted to wrap up the talk part of the workshop, and thank you to Jeanette and Peter for the last two talks. Just for your general information, at 1 o'clock we will start another like get together where our invited speakers will be there, and everybody is welcome to join. We will do that not on Zoom as we originally planned, but on this platform called GatherTown. There's a link on Zulip, how you get there. So if you go to the speaker rooms, then there's a link to the general information where it says how to get in there. I can maybe very quickly illustrate that. So there will be a link that you can open in your web browser. And when it finally opens, then you should type in a password, but apparently I already did that. And then you type in your name. there will show up your video and audio, and then you can join the gathering. Yeah, well, maybe it doesn't work when I'm on Zoom at the same time, but on that platform, what's nice is that everybody can be online in the same room, and we can move between different conversations, and you can always have local conversations as long as you stay close to each other. If you move away, it's a different conversation. Yes, so thank you very much to all of you for participating in our workshop. We three definitely enjoyed it, and I hope all of you also enjoyed it. And we hope to see as many of you after lunch in the social event. And I think the only restriction really was that Jeanette had to go by latest 2 o'clock, and otherwise, I guess we'll see how long people will hang around.
Yeah, Jan will be a bit late, I think, probably around 13:30.
Okay, great. Okay, then I will leave the room open. Feel free to discuss as much as you want.
Thank you
very much to all of you.
Thank you, everybody. See you around.